حمدا لباري الأنام ثم الصلاة والسلام ما ناح في دوح الحمام على الرسول العربي وعلى آله وصحبه ومن تلا من حزبه سبيله في حبه على ممر الحقب. اللهم أن اللسان زلات فاغفر زلات ألسنتنا والكلام سقطات فاغفر سقطات كلامنا، وأنت المثبت فثبت قلوبنا على الطاعة واعصمها من المعصية واجعل لنا في سائر أمورنا خيرا. Today, MOL midterm crash course. This is my first crash course, and I actually recorded this at the end after I recorded everything. The one that reviewed it, Umar Rahman and Abdurrahman Anali, please thank the guys. And let me give you just an overthrow over the whole crash course. So basically, I'm just going to start with cell signaling 1 to 3. I'll just go all of them in just one recording. I think it took 30 minutes. And then molecular techniques 1 to 3, around 1 hour and 10 minutes. Enzymes 1 to 2 with clinical enzymology. This took around 50 minutes. And then insulin glucagon metabolic effects, feed fat cycle, diabetes, 20 minutes each. I'm just telling you the time, so if you're like at the end, near the exam, near the midterm, you just can which one are you weak at and watch it all. And I think the video is around 3 hours 20 to 30 minutes. I did not calculate it as sure, but it's very good, inshallah. I tried to cover everything, and good luck. Now let's start with cell signaling. So now let's start with cell signaling with the GOAT, Dr. Zaid. And I think it's going to be, inshallah, very easy. We'll go through it and you'll understand everything, inshallah. So now let's start with the types of communication between cells. So first we have the autocrine, where the cell basically targets itself. It sends the signal, and then this signal will then initiate a response inside the cell itself. So that's autocrine. Signal cross gap junctions is when a cell sends signals, okay, to another cell, the target cell, through a gap junction. So that we can see mainly in, for example, myocarditis and the gap junctions that they had. And by the way, I could change. Ligand is basically a molecule that initiate a signal, that binds to a receptor and initiate a response. So it's a signal. So when I say ligand, it's basically a signal. And then paracrine is basically when a cell, okay, it will produce a signal or a ligand in the cell matrix or anything, and then that ligand or signal will target another cell nearby in the area of the tissue. And then endocrine is basically I produce a signal or a ligand or even a hormone, and then release it to the blood, and then that ligand would target another tissue that is probably far away through the bloodstream. And now we have membrane versus nuclear receptors. For membrane receptors, you need to know that they are always hydrophilic because it's extracellular, right? It's on the plasma membrane, and the ligand that binds to it is hydrophilic. Okay, it's polar. Why? Because the plasma membrane is hydrophobic, it contains fat. So any hydrophilic molecule cannot pass through the plasma membrane. So what do we do for it so it can send the signal? We make a receptor that's inside the plasma membrane, and then the ligand binds to that receptor, and then there will be a cascade of signaling with amplification of the signal, like this, sends the signal to this, and then that will send the signal to this. You see, the signal is being amplified, and then until there is a response. Okay, but what if we have like a hydrophobic ligand? Okay, it can pass through the plasma membrane. Well, we will have its receptor either intracellularly or even in the nucleus. Okay, it will immediately bind to the receptor. They will form a ligand receptor complex, and then they will bind to the DNA, causing change in gene expression, which will lead to a cellular response. Yes, as you can see, yeah, of course, in membrane receptors, as you can see there, the concentration of the ligand is low because the signal is already amplified. Okay, I don't need high concentrations of the ligand because there are amplification, so the concentration is low. But for nuclear receptors, there's no amplification, so my concentration needs to be high. And then these are examples of the extracellular and intracellular ligands. Unfortunately, you need to memorize them. Okay, but when we get to cell signaling 3, there will be a clearer table. You can understand which one is which easier. But for now, just not these. Okay, just see them, like read them. Pause the video for 5 seconds, and then you will memorize them later in cell signaling 3. And now for our main thing in cell signaling 1, which is the J protein coupled receptor. The J protein is first has an N-terminus, which is extracellular, and that's where my ligand binds. So in GPCR, my ligand binds to the N-terminus. And then I'm going to have 7 alpha helices, 7 transmembrane alpha helices. As you can see, these alpha helices, they span throughout the cell membrane, the plasma membrane. Okay? I know it doesn't look like an alpha helix, but these are alpha helices. If you remember beta and alpha helices in the protein structure, these are alpha helices. And as you can see, they're connected by these loops, ECL, ICL, where they're called intracellular loop, extracellular loop, extracellular. So we have 3 extracellular loops and 3 intracellular loops. And these are stabilized by what? Disulfide bonds. Okay, these are one of the strongest bonds you'll find in the body. And they basically stabilize these loops. So we have 3 extracellular loops, 3 intracellular loops. What stabilizes them? Disulfide bonds. And we also have, this is the large J protein, okay? And then at the C terminus, what we have the complex that binds to this, which is my alpha, beta, and gamma subunits, okay? The alpha subunit is actually the active one. Beta and gamma is mainly structural. So when they are inactive, so when GDP is bound to the alpha subunit, that's inactive. But if I want to activate the J protein, what I'll do, I'll convert GDP into GTP, okay, triphosphate, no diphosphate. When I convert into GTP, the alpha subunit will detach from the beta and gamma and from the whole receptor, and then will initiate a cell response. The beta and gamma will then form a dimer, just structural purposes. We don't know mainly what it does. And these are all in the C terminus. And that is for the large J protein. Sometimes we have a small J protein. So we don't have all these loops and everything. We just have the N-terminus for the ligand, and then we just have this, the RAS, the small J protein, which is basically your alpha subunit. And that's the RAS J protein family. And I will explain it later more in cell signaling 2, but just know small and large J proteins. Yes, this is basically it. And now how does it get activated? I said that it gets activated and becomes GTP. How does that happen? Well, we have an enzyme called guanine exchange factor, okay? For every GPCR, okay, J protein coupled receptor, there is a specific guanine exchange factor, which what? It will convert the GDP into GTP. It will phosphorylate GDP into GTP, activating my alpha subunit, which will initiate the cell response. But you don't want excess activation. Okay, you do your job, then khalas, stop. I don't want it to just stay activated. So what I do, my alpha subunit basically have what we call an intrinsic GTPase activity. So it has something inside it that basically hydrolyze GTP into GDP. And how's that thing stimulated? How's that intrinsic GTPase activity stimulated? By another enzyme called J protein, GTPase activating protein, GAP. Okay, the clothing company, yeah, basically this one. So with GAP, what it does is basically activating the GTPase. Okay, it's GTPase activated protein. It will activate this intrinsic GTPase activity in the alpha subunit, and that will lead to the hydrolysis of GTP into GDP, and the J protein is now returned to its inactive state, and then it will form this complex again. And now we said we have a GPCR family. We have different types of J protein coupled receptors. We have stimulatory and inhibitory. They basically do the same thing, just that inhibitory is the opposite, but same mechanism. We'll explain it later. And then we have GAQ, which activates protein kinase C at the end, protein kinase C. Okay, just know Q with C. And then we have GA12/13, which basically activates protein kinase G. For INS, it activates protein kinase A, okay? The one that we all know about in MOL1, okay? We'll talk about it. And then in Q, it activates C, and then in 12 over 13, it activates protein kinase G. So always protein kinases. So let's start with the stimulatory and inhibitory. First of all, we have the ligand binding, okay, and now we have, of course, the guanine exchange factor, phosphorylating GDP into GTP, activating the alpha subunit. And then the alpha subunit, what it does, it will activate an enzyme called adenyl cyclase, okay, adenyl cyclase. What does adenyl cyclase do? It will convert ATP into another molecule called cyclic AMP, okay, cyclic AMP, CAMP. And then what will CAMP do is will activate protein kinase A, protein kinase A, and then protein kinase A will do all the responses and mediate the response that the ligand wants. But as we said, we always want to limit the activation, right? So what we'll do to inactivate CAMP, we have an enzyme called phosphodiesterase, and basically converts CAMP into 5-AMP, which inactivates it, and now the signaling is now stopped. So phosphodiesterase also controls and prevents excess activation. This is for the stimulatory, okay, GAS. But we have also inhibitory. What does the inhibitory do? Inhibit adenyl cyclase. That's it. Inhibitory will inhibit adenyl cyclase, while stimulatory will activate adenyl cyclase. See how easy it is? And phosphodiesterase will also be a part of the deactivation by converting CAMP into 5-AMP. Now we have some problems, okay, some clinical correlations. So for example, my cholera. So we have cholera, we have excess CFTR channels, we have watery diarrhea. How's it called? Basically, in this stimulatory J protein, what the cholera toxin do is that it inhibits GAP, okay, of that stimulatory J protein. So the alpha subunit is always active, which cause what? Excess cyclic AMP, which will cause a lot of CFTR channels, which will lead to diarrhea, watery diarrhea. And then, so what does, there is another clinical correlation, which is the whooping cough. I think see, I mentioned them here. So the whooping cough, what it does is basically, it will inactivate the inhibitory J protein. What did the inhibitory J protein do? It just inhibits adenyl cyclase. So if I have loss of inhibition to adenyl cyclase, I'll have excess adenyl cyclase activity, I'll have excess CAMP, and now in the lungs, okay, I'll have excess mucus production, which will lead to that whooping cough. And now we have the GAQ and how it leads to protein kinase A activation. So first, as we said, ligand binds and the alpha subunit gets activated, or GDP into GTP. Okay, then what happens? The alpha subunit will then activate an enzyme called phospholipase C, PLC. Okay, remember this. Phospholipase C. Phospholipase C will then go to a molecule called PIP2. PIP2, it's in the plasma membrane. And then it will convert it and break it down into two molecules. Diacylglycerol, DAG for short. يعني لما تقول واحد داق بحريني in Bahrain we say داق يعني crazy guy. Okay, so we have diacylglycerol, DAG, okay, and then we have inositol triphosphate, IP3. Now let's focus on IP3. What IP3 will do? It will go to the endoplasmic reticulum. Remember the thing that needs the nucleus. And what it does, it will cause calcium influx from the endoplasmic reticulum. So now a lot of calcium is now released into the cytoplasm. And calcium with diacylglycerol DAG will activate another protein, which is the protein kinase C. So first we have phospholipase, then PIP2, then we'll have IP3 and DAG. First focus on IP3. IP3 will cause calcium influx, and the calcium with DAG that we got from the breakdown of PIP2, they will both activate protein kinase C. And now we go to the last part of our lecture, which is GPCR activation of guanylate cyclase. So as you can see here, we have GA 12 over 13. This is 12 over 13. When it's activated, okay, the alpha subunit will then activate NO synthase. So what is NO synthase? It's basically, it will produce nitric oxide. It's a gaseous ligand, okay? And the nitric oxide will activate another protein called soluble guanylate cyclase. You remember adenyl cyclase? So this is soluble guanylate cyclase. Adenyl cyclase is for ATP. Soluble guanylate cyclase is for GTP. And then it will convert GTP into cGMP, cyclic GMP, which will then activate protein kinase G. Protein kinase G functions you need to know is for smooth muscle relaxation, platelet inhibition, and change in gene expression. But what you need to know really, smooth muscle relaxation and platelet inhibition. As you know, nitric oxide is a vasodilator, so it makes sense when it activates protein kinase G, it will lead to relaxation and platelet inhibition. And as we said, we always want to prevent excess activation. We have also phosphodiesterase, that protein that we know from before. It will activate cGMP into 5-GMP. And that's the whole lesson. No questions. What is direct signaling? Just pause. Cell targets. The cell target a nearby cell by gap junctions. So we have autocrine, which is the cell target itself, and then we have direct signaling, so through gap junctions. Differentiate between the two. And which of the following GA subunits is responsible for activating guanylyl cyclase? So guanylyl cyclase, guanylate cyclase is the 12/13. So now let's go to cell signaling 2. We'll mainly focus on another membrane receptor, which is tyrosine kinases. Now, these are basically plasma membrane receptors. There are 2 of them, okay? But they're not always together. They are like away, the plasma membrane, but whenever the ligand binds to them, they start to dimerize, okay? Start to dimerize. The tyrosine kinase linked receptors, they start to dimerize. You need to know that they are not dimerized from before, but then after a ligand binds, they start to dimerize, okay? And then they become dimerized. And then what is the activation? What happens? Inside these receptors, there is what we call an intrinsic tyrosine kinase activity. Remember, intrinsic GTPase activity? These have intrinsic tyrosine kinase activity. Now, what they do is basically they will phosphorylate some tyrosine residues, which means just some tyrosine amino acids inside this chain, okay? And this chain will phosphorylate the tyrosine residues on this chain. So it's transphosphorylation, right? And the tyrosine kinase in this chain, the intrinsic tyrosine activity in this chain, will phosphorylate the tyrosine residues on this chain. So I'll phosphorylate you, you phosphorylate me, okay? And then when I have these phosphotyrosine residues, so phosphorylated tyrosines, I'll have some proteins with an SH2 domain. We call these proteins adapter proteins. They have something called an SH2 domain. Like part of it, there is a domain called SH2, which can dock and bind to these phosphotyrosine residues. Now, there are two things you need to know. Some tyrosine kinase receptors are already dimerized from before, okay, which is mainly, we'll talk about insulin and insulin-like growth factor. They're already dimerized from before. And what also you need to know that some of these tyrosine kinase, some of these receptors, they don't have an intrinsic tyrosine kinase domain. Okay, they don't have that activity, intrinsic tyrosine kinase. So what we'll do, we'll have another protein that's not a receptor, it's a non-receptor tyrosine kinase, which goes into this receptor and phosphorylates its tyrosine residues. So 2 things you need to know. So of course, the ligand will bind to the N terminus, okay, extracellularly, while the C domain is for the whole activation, okay? And now we have like the structure of tyrosine kinase receptor. As you can see, insulin and insulin-like growth factor 1, the receptors are already dimerized from the start. And as you can see in the extracellular domain, I have like some different structures. I have cysteine-rich domain. I have immunoglobulin-like domain. I have fibronectin-like domain. Why all these different things? Because I want specificity. I want stabilization. I want structure, right? So I'll have different domains for different purposes. And that's basically it. And now we'll talk about, remember when we told that some of these receptors, they don't have tyrosine kinase activity. These are the proteins that actually phosphorylate them. So what is the structure of these proteins? What are the structure of these tyrosine kinases? First, we have in the N-terminus, okay, there are some myrcylylation, palmitoylation. Don't worry about it. It's just some fancy things to tell you, okay, it's just to make the protein be able
to be bound to the plasma membrane. As you can see, it's like loosely bound. So these things just help to be loosely bound. And then after the N-terminus, I have the SH4 domain. This domain, we don't know what it does mainly, okay? Maybe just linker, but other than that, the function is unknown. And then we have SH3 domain, which is to bind to proline-rich sequence. So if I have any protein, something with a high number of prolines, that sequence, like lots of prolines, SH3 can dock and bind to that. And then we have the SH2 domain, which we already know that basically that domain is the one that binds to my phosphotyrosine residues and the TKRs and the tyrosine kinase receptors. And then we have the SH1, which is my kinase domain. So when I don't have any intrinsic tyrosine kinase activity in my receptor, this is the one that's going to phosphorylate the tyrosine residues. Okay, and what you need to know that in this, there is like tyrosine 530. So that the 530 amino acid, that is tyrosine. Okay, when it's phosphorylated, this whole thing is inactivated. Okay, the SH1 is cannot phosphorylate anything. The whole thing is inactivated. But the moment tyrosine 530 is dephosphorylated, okay, so we remove the phosphate, the whole thing is back to active, and the SH1 domain is now active and can phosphorylate your tyrosine residues. Now there are different proteins with my SH2 domain, the one that can bind and like to the phosphotyrosine residue, and that's what we call an adapter protein. Circ-stat, you know, Circ, that's the one you just talked about. Stat, you'll see. PI3K, you'll see. GRB2, you'll see. But what you just need to know, you remember RASJ protein, okay, the one that we talk about. Just know that it does not have an SH2 domain. It is not an adapter protein, so it can never dock into that tyrosine kinase receptors. That's why now you need to know it. So now we have the signal cascade of the RASJ protein. So first I have the ligand receptor complex, okay, and then now the intrinsic tyrosine activity. I have the phosphorylation, the transphosphorylation of my tyrosine residues, okay. And now I want the RASJ protein to be activated because this is for the RASJ protein. But we said RAS cannot dock into phosphotyrosine residues. So what we'll do is basically we'll have GRB2, which is an adapter protein. As you can see here, GRB2, it's an adapter protein, it has an SH2 domain, okay? It will now dock and binds to these phosphotyrosine residues. And then another protein will bind to it, SOS, okay? Which is basically, you remember guanine exchange factor in the first one, which is the one that converts GDP into GTP in the alpha subunit. Well, SOS is RAS's own GEF, guanine exchange factor. So SOS will then form a complex with GRB2, okay, and binds to it. And now I have GRB2 SOS complex. And then SOS, when it forms this complex, it will activate RAS and then converts GTP into GTP. And then I'll have RAS being activated, okay? And then when RAS is activated, it will activate another protein and phosphorylates it, which is RAF, okay? So RAS will phosphorate RAF. And the RAF will phosphorate another protein, which is MEC. And the MEC will phosphorate another protein, another one, which is ERK. And then ERK will go to the gene, to the DNA, and cause different gene expressions, which will mediate the cell response. So I first have GRP2 docking, then SOS, then RAS being activated, okay? The RAS will phosphorate RAF, RAF will phosphorate another protein called MEC, MEC will phosphorate ERK, and then ERK will do, it's the most downstream and will cause all the activation. So it's the RAS, RAF, MEC, ERK signal cascade pathway. Right? And another thing is the JAK stat. Okay, now we said that there are some receptors that does not have a tyrosine kinase activity, right? This is one of them. So now I have first the ligand. Okay, I have the dimerization, and now I don't have a tyrosine kinase activity. Who will phosphorylate my tyrosine residues? Well, JAK is the hero, and he's the one that's gonna phosphorylate the tyrosine residues. JAK will now is the non-receptor tyrosine kinase. It will phosphorylate the tyrosine residues. Okay. And now what will happen is another protein called STAT. It's not a kinase, but it will basically now dock to the tyrosine residues because it's an adapter protein. And then JAK we'll see these stat proteins that are docked, and they'll say, why not get phosphorylated? Get phosphorylated. Get phosphorylated. So Jack will phosphorylate one here, and the other Jack is probably here will phosphorylate another stat here, and these phosphorylated stats, okay, they will unbind, okay, and they will dimerize with each other, okay, and they will form a dimer. And then after forming a dimer, they'll go to the DNA and cause a difference in gene expression. So that's the Jack stat signaling. And this pathway is used by cytokines like interleukins and hormones like leptin, prolactin. Just have like this knowing, because maybe if he asks about it, maybe okay, because he put it in the slides. Just know interleukin, JAK-STAT. And then we have the PI3K signaling. Okay, how does it work? I have the ligand binding, and then I have the dimerization, the transphosphorylation of the tyrosine residues. Okay, and then these tyrosine residues will activate a protein called PI3K kinase. Okay, PI3K for short. PI3K, what I will do, it will go to PIP2. Remember PIP2 from there? That was converted to DAG and IP3. Well, now PI3K kinase will just phosphorylate it and convert it to PIP3. What will PIP3 do? It will recruit act. What does recruiting mean? Basically, as you can see, PIP3 is near the plasma membrane. Basically, it will just lure act and make it come closer to the plasma membrane to recruit it. And when the act is closer, it will get phosphorylated by another protein, which is called mTORC2 or PDK1. As you can see, PDK1 or mTORC2. Okay, so PIP3 will recruit, come closer, and then what does the job is mTORC2 or PDK1 phosphorylating act. And the phosphorylated act will then phosphorylate another protein called BAD. I don't know these weird names, man. والله اسمها غريبة جدا. Okay, and BAD will then mediate the cell responses. And this PI3K signaling only happens when there is like cell survival, like life or death for the cell. It's like in really tight situations like ischemia or something. So that's where that signaling activated. So as maybe you can think of it like bad, when it's activated, it's like we are in a bad situation. We need to survive. Okay, and then we have insulin signaling, the easiest one of the bunch. Just know that it's already dimerized the receptor, and then when insulin is there, the transphosphorylation will happen, and then it will activate IRS1. Okay, another protein IRS1, and the phosphorylated IRS1 will activate many pathways. It could be PI3K, it could be RAS, it could be even protein kinase A. Okay, and that will lead to cellular responses that insulin do, like cell growth or increased glucose uptake. Enough for questions? Why is it not working? Okay, which of the following domains of circ kinase in the catalytic domain is the catalytic domain? Okay, finally it loaded. Which one is the catalytic domain that phosphorylates everything? SSH1. Which of the following proteins is responsible for directly phosphorylating act during PI3K? So it's the one phosphorylating. It's PDK1. Okay, now the last signaling. This is probably the easiest. Okay, unfortunately, you need to memorize this. Wallah, I know why, but you need to. Okay, I'll try to make it easy. Gaseous hormones, I think it's very easy to know. Nitric oxide, hydrogen sulfide, carbon monoxide. Whenever you feel it's chemistry you're listening to, okay, immediately know that this is a gaseous hormone. Fatty acid derivative, we have prostaglandins and thromboxanes and leukotrienes. Prostaglandins, thromboxanes. Remember, it reminds me of a very dark time. وقت أسود الجاهلية FND. So whenever you feel that you're remembering FND, prostaglandins, thromboxanes, no, that's a fatty acid derivative. And then we have amino acid derivative, epinephrine, dopamine, thyroxine. That reminds me of another block here. ERP. So whenever you see what's like an ERP hormones, it's an amino acid derivative. And then when we have peptide hormones, insulin, glucagon, growth hormone. So this, honestly, I cannot give you something, but you will later in the crash course, you learn about insulin and glucagon in like detail and how they are peptide hormones. They're just made out of amino acids. So insulin, glucagon, and you remember that growth hormone has the same receptor as insulin, just so, okay. So insulin, growth hormone. That's growth factor, but just try to memorize growth hormone is a peptide hormone. And the steroids, whenever you see like cortisol, aldosterone, or any of your sex steroids, immediately it's one of the steroids. And steroid hormones, as I said, we have two types. We have glucocorticoids, and then we have the six hormones: testosterone, estrogen, and progesterone. And for these, now we're talking mainly about steroids, right? Now, steroids doesn't mainly do for ion channel, but ion channel receptors, they initiate the response, right? Like in milliseconds. Immediately you do that, and then immediately it opens, and the response happens in like milliseconds, very fast. But then for membrane receptors, sometimes steroids will go to a membrane receptor. Rarely, but sometimes they do. Okay, and these pathways take like seconds to minutes. Okay, it takes seconds to minutes. But then the one where it's steroids, where it binds to the receptor complex and then go to the DNA and then change gene expression, and then I'll have transcription, and then I'll have post-transcriptional modification, and then I'll have translation, and then I'll have post-translation modification. Woo! hours to days وين طول الحج طول. So ion channel milliseconds, membrane initiated blah 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 blah, membrane initiated steroid signaling seconds to minutes, and nuclear initiated steroid signaling hours to days. And now we need to know the nuclear receptor structure. First we have end domain. Okay, this is the N terminus. لا حول ولا قوة إلا بالله. I know domain. N terminus. تمام. بعدين عندي the variable domain. This is the one, because receptors, they go inside the DNA, they interact with lots of transcription factor. That's the one. The domain one, the closest to the N terminus, that's the one which interacts with all the transcription factors. And then have two is where is my DNA binding domain. It's where the receptor binds to the DNA through what we call a zinc fingers motif. Yes, remember, it's like, think of it, it's like fingers that are holding the DNA, and that's how I remember it. And they are made of zinc. I don't know. Also, zinc is like I think it forms some green. I don't know. Just zinc fingers that hold the DNA. And then we have three domain for dimerization, because as you can see for steroids, you will see that they always form a dimer, right? And it's not the actual steroid that forms like another steroid coming and forming a dimer. No, it's a steroid and a receptor, and another steroid and a receptor, they bind together. Okay, so the receptor of one and the receptor of two, they bind together through the domain 3, which is the dimerization domain. And then we have 4. As you can see, it's closer to the C terminus. So unlike the membrane one, where the ligand binds to the N terminus, closer to the N terminus, the domain for binding here is closer to the C terminus, and that's where the hormone binds. And then we have 5, which is my nuclear localization signal. This is basically a signal that needs to bind to guide the receptor to the nucleus and DNA. For motif a domain, we've said this in MOL 1, but I'll just go over it. Just that motif is a super secondary structure. It's not the tertiary structure of a protein, and it has a structural purpose. It does not have and cannot be independent. But domain is a tertiary structure. It has many functions, and it's very stable independently. So now we have 2 types of steroid intracellular receptors. Now let's go with type 1. Type 1 is always found in the cytoplasm, and it's bound to a heat shock protein, which basically inactivates it. So let's say we have estrogen as an example, okay, and the estrogen receptor now, it's type 1, so it's bound to a heat shock protein, okay? Now when the estrogen comes, it will bind to the its receptor, and when it binds to its receptor, the heat shock protein detaches. And then after it binds to its receptor, another estrogen will bind to its receptor, and then we'll have a homodimer, like dimer of the same thing. So estrogen and receptor, they form a dimer, and then they translocate to the DNA, and they bind to what we call a hormone response element region of the DNA. Okay, so if it's estrogen, it's going to be estrogen response element region of the DNA. Okay, and then that will initiate the cell responses and all that gene expression stuff. As you can see, HRE, response element. And type 1 includes 6 steroids and corticoids and aldosterone. So basically anything coming from your adrenal gland, from your suprarenal glands, adrenal glands, and or the gonads, it's a type 1. It has a type 1 receptor. Now for type 2 receptors, For example, we have vitamin D, vitamin A, thyroxine. Just remember vitamin A and vitamin D, the fat soluble vitamins, and for thyroxine, thyroxine, I don't know. So these type 2 receptors, they're found actually inside the nucleus. Okay, so they're closer to the DNA. So what binds to them to basically deactivate them? What we call a co-repressor. So now let's say vitamin D goes inside the cytoplasm and enter the nucleus, and then what will do? It will bind to its vitamin D receptor, which will lead to the detaching of co-receptor. And then vitamin D, which is with its vitamin D receptor, it will form a heterodimer with another receptor called RXR. XR. I don't know what it stands for. Just know it's RXR. Okay, so it forms a heterodimer. It's not a homodimer like the same thing. It's a heterodimer, which then binds, and they form this heterodimer, and then binds to my vitamin D response element, which will lead now to the gene expression and all that. Now it could be, for example, vitamin A, and it's gonna be retinoic acid receptor, RAR, because vitamin A is retinoic acid, RAR, forming a heterodimer with RXR and does the same thing. But type 2 receptors, they can either form homodimers or heterodimers. It's not like specific. So it can be either or. And yeah, now for questions. Which of the following is a gaseous hormone? So think of chemistry. Which one reminds you of chemistry? كيمياء والقصة مع الغازات ولا هذا. ايش يذكرك بالكيمياء؟ هيدروجين سلفايد. بعدين عندك now we have which of the following receptors forms a heterodimer? Is it testosterone? Is it vitamin D? Is it estrogen? Progesterone? Or corticoid? Final is your answer. And our $1 million winner is B. Easy peasy, lemon squeezy. Now we go to the one all of you are afraid of, molecular techniques. Okay? Molecular techniques, the one that I've heard so much about. People are like afraid about the seniors, they have complained about this for like eternity now. But believe me, this is one of the easiest topics in MOL. And with knowing the buzzwords for every technique and everything, it's gonna be easy as a piece of cake. Inshallah. So first, why do we learn it? We have techniques to diagnose any diseases. You'll see lots of examples for diagnose diseases. Sometimes for treatments, we create like a personalized treatment and all that. But this one, I can't remember the exam, so I'll just skip it. So now we have our first thing. And this is by far, you need to know that this is the first thing that we do and one of the most important things that we do. This is the step before any technique you'll probably see that is happening with DNA, which is restriction endonucleases. Our DNA is huge, very big, billions of bases. We cannot test that DNA in billions of bases. We need to have it in small testable fragments. So how we do that? Through an enzymes that we found in bacteria called restriction endonucleases. Now what they do, these enzymes, okay, or these restriction endonucleases, whenever they see a specific sequence that they are programmed for, when they see that sequence, they just cut. No matter where is the DNA, no matter where. When they see that sequence, I cut. I see that, for example, let's say that the program sequence is GAATTC, for example, here, and GAATTC. This is more of palindrome. I'll explain it. When it ever sees this sequence, it will just go cut, cut, cut. Now, what you might notice about this probe, or about this sequence, is most of the sequences here, when you read it from start to end, you can read it the opposite from the other strand. So, for example, from here, GAATTC. And then when I go to the other strand, it's GAATTC. Same thing. For example, I think here, GGATCC. GGATCC. So what we call that's a palindromic sequence. So palindromic sequence is just a sequence that you can read from the start and from the end the same. And there are two types of cutting, like after cuts. There are what we call sticky end cutting, and then there is like blunt end cutting. So what we call a sticky end is when we have the strands, they're not equal, right? It's like this one strand that's like this, and then a strand that's a bit longer. That's what we call sticky end. So for example, here, you see the restriction nuclease cuts when it reaches the sequence, it cuts right after the G here, right? And the same here, it cuts from here. So now I'm gonna have this part, this cutting part is gonna have one base from here, and then the other strand I'm gonna have, see, a long sequence. And the same thing from here. When I cut, I'm just gonna have one base from here, and then a big base from here. You can see it here, like see when it cuts around here, it's like sticky and it's not equal, right? But here, for example, it's blunted. So for example, yeah, here, GGCC, GGCC, it just cuts at the middle. There's 2, 2, 2, 2, and I will have equal band. Okay, so that's what we call blunted. And this is the step before anything, especially before blots. If it tells you about I'm doing a blot, and that's what's the first step, always restriction endonuclease. Whenever you see it as and they ask about the first step in blotting, and you see cutting the DNA or restriction endonuclease, always put it. Now, this is what we said. Yeah, it cleaves in 5 to 3 direction. Almost everything in the DNA is like in a 5 to 3 direction. But yeah. Now, DNA cloning. Now, for example, I have fragmented my DNA, and I've taken a fragment that it has the gene of interest now. For example, it has the insulin gene. It's just an example, okay? And now I want to amplify that fragment so I can use it in the market. I want to mass reproduce the gene, and then I get a lot of insulin so I can sell it. So what I'll do, I'll cut my DNA through a restriction endonuclease, and then take the fragment that has the DNA that I want, and then I will go to a plasmid, which is a circular DNA inside bacteria, and then I will cut it. I'll cut this plasmid with the same restriction endonuclease, okay? And then what I'll do is I'll take my fragments and I'll take my cleaved plasmid, and then I'll ligate them. I'll put them together through an enzyme called DNA ligase. Now I have this hybrid plasmid with my DNA of interest, right? The fragment that I has and the whole plasmid. And what I'll do, I'll just reintroduce this plasmid into a bacteria. Now, what we know about bacteria reproduce very fast, very fast. They immediately form colonies of millions of cells. It can be one bacteria cell, and you check like 1 day later, it's like 200,000, a million. They reproduce very fast. So why is that good? Because then when it replicates, okay, and reproduces, right, my plasmid will also be replicated. Let me just take this. I don't know, how can I take it? Yeah, okay. You see, my plasmid is now also replicated with it. So now after the bacteria like formed a million copies of my plasmid, what I'll do, I'll kill the bacteria, I'll just slice it. Assalamu alaikum, you did your job. And then I'll take the plasmid. I'll extract all these plasmids that the bacteria mass produced. And then with these plasmids, my hybrid plasmids, I'll cut them back with my restriction endonuclease, and now I'll have this amplified fragment of my DNA, and then I can do, I'll just take it, extract it, and then I'll probably do the transcription, and I have lots of insulin now. So that's basically DNA cloning, but that's the old times. Now they mainly do PCR replication. We'll talk about later. It's just automatic. There is no bacteria, nothing, just PCR. And DNA cloning. Now probes. Now when I say probe, okay, I just mean a molecule with a signal. That's it. When I say a probe, it's a molecule with a signal. Now, for example, when I say a DNA probe, I just mean I have a single-stranded DNA, okay? I have a single-stranded DNA, and I put, maybe I'll produce that probe out of radioactive material. So when it ever hybridizes with the DNA, right, and binds, I can detect it through radioactivity. Or maybe a fluorescence, like light. It produces a light whenever it hybridizes. So, for example, and then I can detect that light. It's just something, a molecule with a signal that I can detect. Now, most of the time, I have like, I want to test if this gene that in my sample is there or not, right? I want to test if it's there or not. So what I'll do, maybe I'll take the mRNA of that gene, okay? and then make its DNA through reverse transcriptase. You know, reverse transcriptase is an enzyme that makes single-stranded DNA from the RNA. Okay, so now I have this complementary DNA for my gene of interest, and then I'll add a signal to it, either radioactivity or fluorescence, whatever, and then I'll add to the sample, and then whenever that DNA, single-strand DNA, binds to my gene because it's complementary to my gene, and now binds to it, I will have a signal that will be sent to me that I can detect. Light, radioactivity, doesn't matter. So that is when I mean probe, because I'm gonna say probe a lot. So I need to understand what does probe mean. Probe means a molecule with a signal. Okay. This is what I said. Yeah. Also, it doesn't need to be like the DNA. It can be like an antibody, because for if I want to detect proteins, how do I do it? It's like antibodies that bind to the protein, which I sometimes call antigen. But basically, antibody is my protein that binds to my protein I want to detect and sends me the signal. I'll explain it now in like I said, enzyme linked immunosortants. Oh my god, I'm just gonna move back forth, right. يلا طيب. Enzyme linked immunosorbent assay, I was calling it ELISA, and it's ELISA from what I've heard. Maybe I'm still pronouncing wrong, but ELISA, oh my god. Anyways, so let's say I want to this is one of the most used assays, by the way, one of the most used, especially clinically. Let's say you have a patient and you want to see if he has HIV. You'll take a blood sample, or I don't know, any sample that has HIV. I don't know. Anyways, a sample from your patient. And what you'll do, you now want to detect HIV. How? HIV is a virus, like you can't just bind to the virus. No. Viruses, pathogens, they always have proteins. And they have like this specific protein just for them, and that we call it an antigen. And what we'll do, we create antibodies that binds to that antigen that is specific to my virus, to my pathogen. So I have antibodies for the protein of HIV, a specific protein of HIV, which now I call antigen. So I have an antibody and it binds to my target antigen. Now, how does ELISA work? Basically, first I'm gonna have a plate. Okay, I'll put all the buffers, everything there in that plate. And then I will put a capture antibody. Now, this capture antibody will be immobilized by the plate. I'll put the antibody and then it will be immobilized in the plate, okay? So now it's just stick it to the plate. And now I will add my sample. Now let's say my sample, the patient really have HIV. What will happen? The antigen of the HIV, so the protein of the HIV, which is my target antigen, it will bind to that capture antibody, because the capture antibody is complementary to that antigen. It will bind to it. So when it binds to the antigen, it will be immobilized too. So now I have immobilized antibody, and now above it I have the antigen. Okay, so antibody and then the antigen, and they're both immobilized. What I'll do is I'll add another antibody, okay? And that antibody will bind to my target antigen again. So it's like one antibody here, the antigen, and an antibody above it. Now, what will happen is that this antigen, when it's ever the second antigen, my detection antigen, my detection antibody, okay, whenever it binds to my target antigen, what we'll do is it will send the signal, okay, I am bound to the antigen, do the enzyme-linked reaction, which what produces a signal. Okay, it will do an enzyme-linked reaction, which can produce then a light, something that we can detect, a signal. And that's what we call ELISA. And it's basically the target is the antigen is sandwiched. It's immobilized first by my capture antibody, and then I put it, right, and now it's immobilized, and then I put another antibody above it, which will send a signal. So this antibody is the same as this one, but basically this is immobilized in the plate, but this is not, but it has an enzyme-linked reaction happening. Now there is another type of ELISA, maybe he'll ask you about it, is that rather than the enzyme-linked reaction happening here, okay, I'll just have the capture antibody, then the antigen, right? Then I'll have another antibody to that antigen and binds to it, And the one I'll have another antibody, another one. I'll have another antibody binding to my second antibody. So it's anti-human antibody. And whenever it binds to that second antibody, it will send the signal. So just to make it easier, whenever you see antigen antibody testing and you see like more than one antibody, it is 100% he's probably talking about ELISA. And yeah, that's basically it. But what you need to know that ELISA is just a screening test. What I mean by screening test, that it's not like a confirmation test. Okay, when I see a positive result, he has HIV. No, it's just a screening test because it has a 20% chance of false positives. So whenever I have a positive result, I don't say, okay, you have HIV. I'm sorry to you. No, what you'll do is you will do a confirmation test, which is by a technique called Western blot. We'll talk about it later, but it's more of confirmation to ELISA. ELISA is your first test that you do, is the screening. Then that is the confirmation whenever you have a positive result. So this is what we talked about basically for HIV and the positive and negative results. Like see, I have positive, and the negative. So if he is negative, خلاص, I don't need to do Western blot. He's 100% negative. But sometimes I'll have a positive result by ELISA, but then it is negative with Western blot. That means it's a false positive. But it can be also positive in ELISA. And then when I do the Western blot, the confirmation, it is actually positive. You have HIV. Okay, I'm just moving on these things. Okay, now fish. Oh my God, I'm actually moving all throughout the slides. Okay, now fish basically is the only test you'll see here and the only technique that you'll see here where I'm doing it inside the cell. Okay, I'm not extracting any DNA. I'm not doing any extraction. I'll just have the sample. I'll do my technique. I'll just add something and I'll see for the results. It's the only one that is in situ inside the cell, and it's mainly for what? For chromosome activity. I want to see how the chromosomes are arranged. I can do it for like Down syndrome. I want to see, is there a third chromosome here? And yeah, so what is basically fish? Fluorescent in situ hybridization. I'll have my sample, okay, and then I'll just, maybe I want to detect, okay, is that specific gene there, right, in my sample? So I'll just have the sample. I'll add a probe that is complementary to my gene and add to the sample. That's it. And if that gene is there, I'll just see the signal detection. I'll detect the signal that the probe will send. I already hybridized to the gene. The gene is here, and I can see it. See, like there's lots of red. But if you see like one or two red, that's probably a negative result. Okay. But if you see many, okay, this is positive. Now, when do we use it mainly? He will probably only talk about breast cancer. So when we have breast cancer, right? I want to see if that cancer can respond to chemotherapy. We need to see if that cancer sample has a high expression of a gene called HERS2. And if that cancer sample has high expression of that gene, that means, okay, we can do chemotherapy. The cancer will probably respond to the chemotherapy and die. But if it's not, and there is no high expression of that gene, then your patient probably cannot respond to chemotherapy. That is basically it. So I'll just have a probe that is complementary to my gene, HERS2, add sample. And if it's a positive result, okay, there is a lot of expression, this patient will respond to chemotherapy. And if it's a negative result, unfortunately, this patient will not be able to respond to chemotherapy. Yes, HERS2, it can be, as I said, for Down syndrome. Now, dot plots. Okay, dot plots is you extract DNA. So first you need to know it's not in situ. You extract the DNA, okay, and then what I'll do, I want to see if, for example, for cystic fibrosis, is my patient homozygous normal? I have three situations. Either my patient is homozygous normal for the CFTR gene, so he's fully doesn't have the disease. Is he heterozygous? So he has one mutated gene one normal gene, so he's a carrier, or he's fully affected, like two mutant genes. So what I'll do is I'll extract the DNA, and then I'll make a probe which is made out of I cannot, what is called? Allele specific oligonucleotide. So because in our DNA, in every gene, we have two alleles, right? One from our father, one from our mother. So what I'll do, I'll make a probe, okay, that is allele specific for the normal allele. One probe is for the normal allele, and one probe is for the mutant allele. So one probe that will bind to my mutant allele, and one probe will bind to my normal allele. So, and then I'll just add it and see what's happening. So when I add my normal allele, I see a positive result. So that allele is there. He has a normal a normal variation of the gene. Okay, but then when I add the mutant allele, okay, I see it's a negative result. So if he's positive for the normal and negative for the mutant gene, that means he is homozygous normal. But let's see, for example, this one, carrier. Basically, when I add the probe for the normal allele, I'm going to see a positive result. And when I added a probe for my mutant allele, I'm going to see also a positive result. So both alleles are there. That means he's heterozygous. But if I add the normal allele and it's a negative result, and then I add the mutant allele and it's a positive result, that means both of his alleles are mutant variations, which means he's unfortunately a cystic fibrosis patient. Now, the use of dot plots, he'll only probably ask about cystic fibrosis. And what you need to know that dot plots are very cheap. And as you can see, it's simultaneous. I can just take the sample and then I'll add the normal allele in one part, and then I'll add the mutant allele in a second test, right? And it's very simultaneous. So whenever you see cheap, simultaneous, and it's for DNA testing, for alleles and that, and especially if you see allele-specific oligonucleotides, immediately dot plots. Okay, now gel electrophoresis. Now let's say you did the restriction endonuclease, you have the fragment, everything, and you want to see if that fragment is there. You'll not do a dot plot, you'll not do fish, nothing. You just want to see if that fragment is there. You use gel electrophoresis. Now, how does gel electrophoresis work? First, I have a gel that allows my sample to move in, okay? And then I'll have this gel to have an electric current. So I'll have a cathode at one side, and a cathode is a negative charged electrode which attract positive substances. And I'll have on the other side, okay, on the other side, I'm gonna have an anode which is a positive electrode which attracts my negative molecules. And we already know that DNA, RNA are negative molecules. So what I'll do, I'll put my DNA sample near the cathode, because I know it's not going to go to the cathode, it's going to go to the anode. And I want to test how far it moves to the anode. I put it inside to the cathode, and now it's electric. I have the electric current running and everything, and I just wait. I'll probably add ethidium bromide or to my DNA, or any signal that tells me, oh, the DNA is moving, by the way. Tell me, where is the DNA actually moved? Because the DNA does not have a light. So I need to add a probe that tells me where have the DNA moved. And mainly in gel electrophoresis, because now I have many fragments of DNA and I'll put it in the gel electrophoresis, right? The larger fragments do not move as much as your smaller fragments. Think of it like the larger fragments are old people, and the small fragments are like the young people. So young people, okay, I can run faster and I can go closer to the anode, while the larger fragments, the older people, they will just run out of breath and then stop midway or really far away from the anode. And that is basically gel electrophoresis. Now we can also use it for proteins, but it's another type of gel electrophoresis. It's even vertical. This is more horizontal, but we'll talk about it later. Now we have two types of gel electrophoresis, and just depends on your gel. Okay, which this one is an agarose, and the second one is going to be polycrylamide. There is another one, phosphate, but don't worry about it. Let's focus on agarose. Now, agarose is when I want to test a DNA fragment that is 200 to 20,000 bases. If I have a fragment that is between 200 to 20,000 kilobases, I'll do a gel electrophoresis to detect it, if it's there or not. Okay? And how do I determine the concentration of agarose? Because the concentration of agarose actually is very important to the fragment size that you're testing. Because the smaller the concentration of agarose, the easier it is for my larger fragments to move. Okay, think of the concentration of the agarose is like rocks and all that, and if there is less of it, the road is more smooth, so the larger fragments can move more easier, right? So if I, for example, I'm testing 15,000 base fragments, I want to detect it. I'll probably use the smallest concentration, like 2%, 1%, 0.5%. I'll probably test it so I can see that larger fragment moving, and I can differentiate between, for example, between the 17,000 and the 20,000, right? Because now I can have detection between them. But then my smaller fragments, they will move all far away, right? They'll move all closer together, that they will join together. But let's, for example, I want to detect a fragment that's like 200 bases. So it's a very small fragment. I want to increase the concentration. So now I'll have my larger fragments. They'll form like a one band. That's probably 15 with a 20,000. But then my smaller bands will start to have space between each other and form like different bases. And now I can see, okay, this is 50 bases. This is, for example, 120, and this is like 200. And now, okay, this is my band. It's here. But for example, when it's like real concentration, the 50 will join with the 60 and the 70 and the 100, they'll join together because they all move the whole distance. So the concentration of your agarose is really important to determine like which fragment are you trying to detect. The larger the fragment, the lesser the concentration. The smaller the fragment, the better it is to increase the concentration of your agarose. Now, for polyacrylamide gel electrophoresis, we also use it for DNA, okay? But it's only when the DNA is less than 200 bases. And it's mainly used for what? Sequencing the DNA. Why? You'll ask me why. Because polyacrylamide gel electrophoresis is so sensitive and specific that if I have, for example, a 200 base fragment and I have a 199 fragment, if it's in gel electrophoresis, they will both be in the same band. It will not differentiate between them. But for polyacrylamide page, it will have, the 200 will have one band and the 199 base will have another band. So you can easily detect like the number of bases in each band exactly. So it can resolve a one nuclear difference, which makes it very useful when we're doing sequencing of the DNA. We'll talk about sequencing, it's at the end. But yeah, yeah, polyacrylamide for the gel needs something called bisacrylamide, cross-linking again. Just but what you need that to know is bisacrylamide is needed for polyacrylamide, and that it is a neurotoxin, and that polyacrylamide, as you can see, the advantages and disadvantages, it is as I said, it has high degree of resolving power. It can be used for sequencing. Now, don't mind this. The doctor specifically said 200, so don't mind the 500 here. But the disadvantages is that it, as we said, that it has neurotoxins, and its gel is very difficult to handle. It's very sensitive, that it can break easily, and it will create a mess. So it is very difficult to handle. For pulse field gel electrophoresis, as we said, it's mainly for large fragments like bacterial chromosome, 20,000 kilobases. Bacteria doesn't have chromosomes. Why did I put that? Anyways, like bacteria DNA, like large fragments. I'll just edit it later in the slides, but bacteria, I don't think they have chromosomes. So when we have large fragments, I'll put it in pulse field, and because you see pulse field has multiple cathodes and anodes, the DNA will just go zigzag motion, and like that. And it's not used a lot nowadays. Now for the blotting. The one that I've been talking about, oh, Western blot, Southern blot. What is blotting? Blotting is mainly, I'll just explain it, what's the confirmation test. And when I say, for example, Southern blot, I mean a confirmation test for DNA. And when I say a Northern blot, it's a confirmation test for RNA. And when I say Western blot, it's a confirmation test for proteins. So I want you to think more of it as like the central dogma. You know, when we want to create a protein or something, first gene expression for DNA, and then transcription and making RNA, and then making the protein. West, so it's like DNA, then we go to RNA, then okay, it's actually opposite. Then I have in the Western side, I have my protein. So DNA, RNA, protein. So now northern and southern blots, DNA and RNA has almost the same thing. So what I'll do, I'll basically do the restriction endonuclease and cut the fragments of the DNA. I'll do my gel electrophoresis and everything, but then I want now to put probes and detect like a specific band, okay, and that unfortunately the gel matrix for agarose or polyclaramide, as we said, they're very difficult to handle. They cannot they cannot how can I explain it? They cannot. I have a brain freeze now. Basically, when I put chemicals, they just cannot handle it. They will destroy the DNA, will go everywhere. They just cannot handle it. So what I need to have my DNA results, like all the gel electrophoresis and the traveling of my DNA, to go to another membrane, to another paper, okay, where I can do all the stuff of detection and washing and all that. So what I'll do, I'll first have my gel, okay, and put it here, okay, and then I'll put a nitrocellulose paper above it. The nitrocellulose paper is the one where I want to do all the testing because it can handle it. It is a buffer. It can handle all the things that I'm going to do, all the chemicals I'm going to add for the detection and all that. So I'll put it here, okay, and the DNA will move to that nitrocellulose paper by passive diffusion. It'll just move through passive diffusion and go up, okay. But I don't want the DNA to move further than the nitrocellulose paper. I want to trap it there. So what I'll do, I'll add like a lot and a lot and a lot of paper towels, or which, and then I press it on the nitrocellulose. So when the DNA is okay moving, and now it moves to the nitrocellulose, and then when it moves, it wants to even move further. There is the paper towels that are inhibiting it and just trapping it into nitrocellulose, because now the DNA cannot move in the paper towels, so it'll just be okay, I'm trapped in the nitrocellulose paper. So now I have nitrocellulose paper, or it can be a nylon paper, modified nylon paper. And after that, okay, I take my nitrocellulose paper with my DNA results now that it's not in the gel anymore, and I can do all that stuff. I can add all the chemicals. I can put the probes, the signals that I want. Okay, just to take one single fragment, and then I'll have the detection. Okay, washing, and then I'll do the detection. Now the probes, okay, I add a specific route for a specific fragment, and then I can detect it. Like, see, this is a radioactive one with radio waves. I can see, okay, this is the band that I want. This is the band. You see how that band thickens? That means that the probe was sent there, and it's sending us a signal. Oh, this is your band, by the way. This is your fragment that you want. And this is the same for chromogenic, so it's with light. As you can see, this is the bands that are thickening. That means that the fragment that we want is there. So this is for south and north plotting, which is for DNA and RNA. This is my confirmation test. So if I see that positive result here, that means that gene I'm talking about is here, or that RNA that I'm talking about is here. But what you need to know, that northern blotting is rarely used because RNA is very unstable, very, very unstable. So it's not used a lot. It's mainly southern blotting, just for your information, because Al might give you a question for understanding and that, and tells you about northern blotting. Northern blotting and RNA is very, very unstable. So yeah, these are all the steps. Yeah, you need to denature DNA, of course, with alkali and cut with restriction nuclease, as I said. And this is basically all what I said. And then Western blotting. Now, Western, we said it's for proteins. Now, what I'll do now, of course, I'm not gonna cut proteins with restriction in the nuclease. It's for DNA, so I'm not cutting proteins. But sometimes I want to do it for molecular weight. I want to differentiate between proteins by molecular weight. I know that my specific protein will be that amount of weight. And with gel electrophoresis, we said for DNA fragments, they move upon their size, right? The smaller fragments will move further, while the larger fragments will move less. Same thing for proteins, but that proteins with smaller molecular weight will move further, but proteins that are heavier have larger molecular weight, they will move less. That's basically it. But the thing is with proteins is they have lots of amino acids that can be positively charged. There are some amino acids that are negatively charged, and that's not good. Especially when I do the gel electrophoresis, I want my proteins to be uniformly charged. I want them to be one single charge. So what I'll do, I'll add something called SDS, what it's sodium dodecyl sulfate, just know it's SDS, okay? And I'll also heat it, okay? Heating and mid-carb-ethanol will make sure that the protein is not in starch structure, it's just primary structure, because I don't want any of their intermolecular bonds or nothing. I just want it purely in its primary structure, just the sequence. And what will the SDS do is make sure that the protein is fully negative. And it will just change the whole protein charge into a fully negatively charged. And now I can have this protein and now add it to what gel electrophoresis we said, specific for proteins, polyacrylamide only. When I do gel electrophoresis for proteins, I'm only using polyacrylamide, which is the vertical one. Okay, so now I have these proteins that have been denatured and now they're fully negative, and now I put it in the polyacrylamide gel, and of course they're gonna move through to the anode because now they're fully negative, so they move to the anode, to the positive charge, and as I said, they will move with their molecular weight. So now you see this small protein, right? It is the lightest. It will move the furthest. A. This medium protein, okay, when now I heat it and everything on SDS, it will move medium because it's the medium one, right? And then the largest one, the biggest one with the largest molecular weight, it will move the least. Okay, and now after I do the gel electrophoresis with polyacrylamide, I'll do the same thing. I'll add it to nitrocellulose paper, the whole thing, and then I take the nitrocellulose paper or my nylon paper, and then I add all my probes there. The signals is probably going to be antibodies, okay, because it's proteins, and then I detect either radioactively, it can be a radioactive signal, or maybe a chromogenic signal, so light. And as you can see, this thickened band, that means, okay, the protein that I'm looking for is here. And that's Western blotting. So for example, if I have ELISA, right, at the start, positive result, then I'll add my sample and do all the protein extraction, and I do all these steps, and then add the probe, okay, and the antibody for the HIV antigen, and try to detect it. And if it's a positive result, this patient unfortunately has HIV. So yes, as we said, it's for protein confirmation. Now SMP or LFP. This sounds like gibberish, and I know. SMP means single nucleotide polymorphism, which basically means a single change of DNA base. So for example, in one random location of my DNA, I have AATCC. But for example, Umar Uthman will have AAACC. So rather than having a T, I have a T, he has an A, and that's an SMP. And that's normal. Everyone has SMPs, and it's actually we have almost everyone has 15 million SMPs in his DNA. So I have 15 million different bases than the other guy, and the other guy has 50 million bases different than some other guy, and that's normal. But when can be SMP like be bad? If it's inside the gene and it causes a change in the protein that will be formed, and that's a big, very big problem. And what is RFLP? Basically, I never memorize the name. Restriction fragment length polymorphism. Of course, you don't need to memorize the names. Believe me, it's okay. So, let's say, for example, I have this gene, okay, and in the normal gene, the section endonuclease will cut before it, okay, and at the end. I'll just create this, for example, 100 base fragment, okay. But now I have an SMP inside the gene which may create what? A restriction site for that endonuclease in the middle of it. So rather than the restriction endonuclease cutting at the start and at the end, it will have another new site at the middle and will cut there. Okay, and rather than having, for example, I have one that is 100 base fragment, I'll have two fragments which could be 70 and 30, for example. Okay, and another thing is that maybe at the restriction endonuclease site, right, the SMP happen at one of these sites and will change it. So now, as you can see, when the restriction endonuclease comes and tries to cut, okay, it doesn't see it, so it will not cut. So now I have a larger fragment, and that's what we call restriction fragment length polymorphism, and this can be used in diagnostics. So now, how can it be used clinically? We use it mainly for PKU. It's a disease that we use for like inborn errors of metabolism. We need to know it immediately, because if we don't detect PKU early, this could cause retardation, and for sickle cell anemia. So you need to know like what is happening here. The specific examples for PKU and sickle cell. Now for PKU, the mutant gene, as you can see, this is the normal sites, right? Normal sites for the restriction endonuclease. But then when there is a mutation here, an SCP here, and there is a mutation, it will create a new site for the restriction endonuclease, and it will cut, which will cause a smaller fragment. Okay, see, smaller fragment. Rather than cutting here, it will cut here, and I'll have a smaller fragment. So when I want to detect if someone has the disease or not, I'll just do the restriction endonuclease, and I'll do the whole southern blotting and detect the band. So if someone is normal, he'll have one thick band, okay, of the larger fragment, which is the normal gene. If someone is homozygous mutant, so he has two of his alleles are mutant, he'll be only having one band of the smaller fragment. As you can see, it travels further in electrophoresis. So only one band of the smaller fragment. But what if someone is heterozygous? He'll have two, but it's thin bands. So one band which represent my normal gene, as you can see, it's because it's bigger, and one that thin band that represents my mutant gene. So that is for PKU. What about sickle cell anemia? Now we know the sickle cell anemia and the one base mutation and all that. And what we'll do is changing, and this mutation will cause the absence of a site for a normal restriction endonuclease. So normally, okay, the restriction endonuclease cuts here and then cuts here at the normal gene, okay, in sickle cell. But because there is a mutation here, as CAG becomes ZAG, okay, what will happen is that site will disappear, and the restriction endonuclease will cut there for the mutant. So now the mutant has a larger fragment than my normal gene. So it's the opposite of PKU. So if I'm homozygous normal, AA, I'll only have the smaller fragment. Okay, thick band of my small fragment. It travels further, so my small fragment is here. If I'm homozygous mutant, I'll only have one thick band of my larger fragment. It travels less, so it's the larger fragment. And if I'm heterozygous, I'll have two bands of just one that is normal and one that is my mutant. So in PKU, the mutation will cause a what? A smaller fragment. But in sickle cell anemia, the mutation will cause a larger fragment. That's what I need to memorize. And yeah, okay. And now VNTRs and DNA fingerprinting. VNTRs are variable number of tandem repeats. What does tandem repeats mean? At some specific locations of our DNA, we have just repetition of bases. So maybe at this chromosome 11, I don't know, locus, I don't know where, I'll, for example, have 8 repetitions of 80. 80, 80, 80, 80, 80, 80, 80, 80. But maybe Abdurrahman Al-Anani, okay, at that specific location, he'll not have 8 repetitions. He'll have 4 repetitions. He'll have 80, 80, 80. So why are we talking about? Because these tandem repeats are really important for paternity testing and DNA fingerprinting. So, for example, your VNTRs, your tandem repeats, half of them you get them from your mother, and half of them you get from your father, right? Paternal and maternal chromosomes, alleles. So what I'll do, I'll cut fragments, okay, with restriction and deglaze. I'll cut and I'll just have the fragments with my VNTRs, and I'll do the gel electrophoresis and everything. So, and now I have this child, and then I see the mother, right? And then, of course, the child will have this band from his mother, okay? And then, okay, this band, he does not have it from his mother, so he took it from his father. This tandem repeat is from his father. And now I see which one match, which one match, which one match, which one match. B and D, okay? And then I see 3, okay, he took it from his mother, so it's not from the father's. And then I see 4, oh, 4 is from his father. So now it's either B or D, right? So now I see, oh, B has it, but D doesn't. He did not give him that tandem repeat. That means that B is the father because his tandem repeats, okay, matches the missing tandem repeats of the child that's not from the mother. See, so half of them, 1 is from the mother, 2, and then 2 is from the father, 3 is from the mother, 4 is from the father, 5 is from the mother, and 6 is from the father. And that's how paternity testings happen. I see the tandem repeats of the mother and the child, and I see the missing tandem repeats, and then I will compare it to the tandem repeats of my suspected father. If they match, that means that he is the father. And we do the same thing for DNA fingerprinting. If I have like a DNA sample from a crime scene, okay, I'll do the gel electrophoresis and just have the tandem repeats. And then, for example, my suspect, okay, and then I'll compare the tandem repeats. If they match, that means that was his DNA. If they don't match, that means it's another person's DNA. Now, polymorphism, polymerase chain reaction, or almost in the end, inshallah. As we said, for DNA cloning, okay, there is another and new one, which is polymerase chain reaction. Basically, I'll have a DNA and I just want to amplify it. I want to have larger amount. So whenever you see questions and you see amplification, it's probably talking about PCR. Okay, so what does PCR do? We have 3 steps in PCR. First, I denature my sample and I make it single-stranded DNA. And that is in high temperatures like 90, 94. And then after that, I will cool it into another phase, which is my annealing phase. And then it will be into 60, 64, where my primers, my forward and reverse primers, can bind to the DNA, okay, to guide then my DNA polymerase to start amplifying the DNA. And then after annealing and the primers, okay, are already bounded, I heat it again into 70 to 72 degrees Celsius, which is my elongation phase. And then I add my specific DNA polymerase, which is called Taq polymerase. Why Taq polymerase? Because Taq polymerase is DNA polymerase that can operate at very high temperatures, 70, 72. Your normal DNA optimal temperature is at 40, 35 to 40. But after that, denatures. But I need a thermophilic DNA polymerase so it can withstand all the heat when I do the denaturing of the DNA and all that. So I need a thermophilic enzyme. Okay, so why primers? Okay, I have 2 primers, forward and reverse. Why? Because I don't want to copy the whole DNA. I just want to copy a specific and amplify a specific part of my DNA. Okay, a specific part. So I'll have primer at one end of my fragment that I want, and another primer at another end. So they're basically limiting the polymerization at this fragment only, and the DNA polymerase will only amplify that. So I have forward and reverse primers. And that's basically PCR. Of course, you need to add with the PCR your free nucleotides. Otherwise, how would the DNA polymerase be able to put the DNA where it will get its nucleotides? So you need to put a lot of free nucleotides within DNA. And this whole thing happens in a machine called the thermal cycler, which really just controls the temperature. So you need to add in it the DNA sample, you need to add the Taq polymerase, the free nucleotides, the thing that we did in the lab. So as you can see, these are the temperatures. The perfect one is 94 for denaturation, then 60 for annealing, for primer binding, and 70 for the elongation, where the Taq polymerase start working. And you can see like different temperatures really affect the results, because if it's different temperature, then I'll have different applications. It's going to be weird. As you can see, Hail Mary, like uncontrolled results. So my perfect is 94, 60, 72. And my primers are along like 20, 22 nucleotides, the forward and reverse. So now we have PCR diagnostics. Let's say I've already amplified the sample that I want. Okay, for example, this the virus, EBV virus. Okay, I've done, and now I want to see if he actually has. This is what we use for COVID, by the way. That's how we did how they did COVID. I think the confirmation test for COVID. So first, I'm gonna have a negative blank. What is a negative blank? I'll add for it. I'll have a sample. Okay, I'll just add the Taq polymerase. I'll add the free nucleotides and everything, but I'll not add my DNA sample. Why? Because I want to check for contamination. Because if I don't add my DNA sample, there should be no amplification happening. So this should be empty. But if the negative blank, okay, the negative band has anything, that means I have contamination. What about the positive blank, right? The positive blank looks, for example, I'm testing this EBV virus fragment, right? A specific fragment from that EBV virus. And I know that fragment is around 500 nucleotides. So what I'll do is in the positive one, okay, I'll add an already known 500 nucleotide sequence base and I'll add it so I can compare. If this is like going here, that means it's not a 500 sequence, it's not my my band that I was looking for. It's not the fragment that I'm looking for. But if it matches, it means, okay, it is actually the same amount of bases, so it is probably my fragment. This is actually gel electrophoresis, by the way. And there's specimen 1, specimen 2. Now, what is B-actin? B-actin is basically a housekeeping gene that is found in all cells of your DNA, okay? And we test for it because we want to see the quality of the DNA that we have extracted. If the B-actin gene, okay, after I do the gel electrophoresis and it shows that it's there, that means, okay, your DNA quality is good. But if the B-actin gene, and when I do the gel electrophoresis and it's not there, okay, I got positive result from here, but I don't have a positive result from the specimen for the B-actin gene, the DNA extraction quality is very bad. Like, how does that cell has virus but does not have the B-actin gene? That probably means that from wherever I extracted, it's probably a dead cell or just a very bad quality of DNA. So B-actin is more for quality, positive is for comparison, and negative is for contamination. Like, you want to see if there's contamination or not. So this is PCR RFLP. As you remember, the restriction fragment length polymorphism. Oh, I memorized it. Okay. So this is basically it. What I'll do is, rather than doing the PCR, don't worry. Okay. So now I have the normal CFTR gene. I have my forward and reverse primers. Okay. And I'll just amplify it. And we know that in CFTR, the cystic fibrosis, what happens? We have a 3-base deletion. Okay, so my mutant gene is smaller than my my normal gene. It's smaller, so it can travel further in gel electrophoresis. So what I'll do PCR amplification of the of that, or just the gene, okay, for forward and reverse primers, as you can see, and just amplifying the gene. And if it's a normal CFTR gene and a homozygous normal, I'll just have the larger gene, as you can see in gel electrophoresis, only one band of the larger gene. If it's heterozygous, so I have one normal and one mutant gene, I'll see one band of the normal 63 base normal gene, and then I'll have one thin band also of my mutant 60 base gene allele. And if it's just fully hit a homozygous mutant, I'll just have one thick band of the smaller fragment with the 60 bases, which means he has CFTR. I don't know why I put RFLP. It's not RFLP, it's just PCR. Okay. PCR diagnostics, yeah. And now quantitative PCR. Okay. quantitative PCR. Now we already know about PCR and how it works and everything, the amplification. But if I want to see how much initially of the disease is there, okay, I don't want to only detect if it's there, I want to see how much of it is there. So what I'll do, okay, I have the sample and everything, and then I'll add a probe, okay, a probe that binds to my sample, to that gene or something. Let's say, for example, it's HIV, and I'll add a probe that binds to for my HIV, the HIV genetic material. Or for example, I have bacteria, right? I'll see how much of the bacteria is there. So I'll take the sample and I'll add a probe that will hybridize and bind to my bacterial DNA. And then if there is a large amount, okay, the probe when it binds to the DNA, okay, it will send a signal. It will send a signal, and I'll just keep amplification with PCR, quantitative PCR. I'll just keep amplification until I reach a threshold of fluorescence, of detection, of signal. Okay, it's reached like, okay, I've reached that amount of radioactivity. Okay, خلاص, I've reached the threshold. There is enough of probe that I can detect. And I want to see how much cycles it took for my probe to reach the threshold of signal, okay, fluorescence or radioactivity or that. Now, if there is already a large amount, okay, it will not take a lot of cycles for my probe to reach this threshold. خلاص, there is already a large amount. The probe is already binding with a large amount. It's already close to the threshold. Maybe 1 to 2 cycles. I've already have a huge amount now, and with my probe binding to this huge amount, it will send a huge amount of signaling, which is way above the threshold, and in very small amount of cycles, maybe 1 or 2 cycles. But if the amount of the disease of the bacteria is few, okay, I'll have the probe signaling, but it's just very minimal. But then after 2, 3, 4, maybe 10, 15, okay, after like many cycles, it will reach the threshold. So the cycle number, how much cycles it needed for it to reach the threshold, is inversely proportional to how much there is initially. Okay, so if there is a lot initially, if there is a lot of the bacteria or something of the bacterial DNA in my sample initially, it will take less cycles to reach the threshold. And if it's only a few, then it will take a lot of cycles, way more cycles, to reach that specific threshold. And this is for the disease progression. Okay, for example, I want to see how much bacteria is there. Okay, in that patient sample, he's maybe he has E. coli or something. I'll take the sample. I want to see how much E. coli he has in this sample, and then I add the probe for the E. coli DNA and everything. And it's just a few cycles. He is in a very progressed state of E. coli infection. He probably have sepsis, unfortunately. But if it's only small amount of the E. coli, it will take a lot of cycles for it to reach the threshold, which means he's at an early stage of the disease, and now I can attack it with antibiotics, something like that. So that is quantitative PCR. Now there are 2 types of PCR probes. This is the cyber green. It's a very expensive probe. It's not used mainly. What I'll do is basically when the PCR and now it's trying to amplify, and now when it sees the specific gene, okay, the gene of interest that I'm trying to detect, and when it sees it's become double-stranded, it will bind to it. So cyber green only binds to the double-stranded DNA of the gene of interest. Only double-stranded DNA. Okay, it does not bind when it's single-stranded. No, it only binds when it's double-stranded. So, okay, now I have amplification, and now I have the gene of interest. Now it's double-stranded. The cyber green will bind to it and send the signal and emit the light, okay? And another quantitative PCR probe is TACMAN. This is the one that we mainly use, and it's way cheaper. So basically, now after the denaturation and now the annealing, okay, I have the forward and reverse primers, and then I'll have my probe, okay, at the middle, and it's binding to single-stranded DNA, okay?
And that probe is made out of two things. The R, which is for the signaling, and a quencher, a reporter, okay, which sends the signal, and a quencher. The quencher is basically stopping the reporter from sending a signal, because if I add it and they're just sending the signal without hybridizing to anything, it's not indicating to me that, okay, the gene of interest is just sending signals like this, but it's not hybridizing to anything. So what I'll do is I'll have it hybridized to my DNA, okay? And now I have a quencher that says, okay, do not send the signal, خلاص? But then I have the forward and reverse, okay? And then the Taq polymerase start polymerizing and elongating the DNA. And then when it sees this probe, what it'll do, just break it, get out of the way. And when it breaks it, when through the elongation, what will happen? It will break the quencher from the reporter. And this breakage will send the signal. Now the reporter is alone from the quencher, and it will send the signal, oh, I have been broken now. That means that the PCR is amplifying that specific gene. Okay, here is the signal. And that is TACMAN. Now, also PCR for pre-implantation and prenatal diagnostics. For example, sometimes before birth, you would see what genetic diseases the fetus have and all that. So at 10 weeks, you can do like a chronic villus sampling. At 16, amniocentesis. I don't think he'll ask about it. No way. I think he skipped it. But you just need to know that there is for pre-implantation and prenatal. Okay, I take a sample for prenatal. I take a sample and I now do maybe fish. I'll do maybe dot plotting for certain diseases to see what disease does he have. And for pre-implantation, with IVF, and I have this fertilized now, and I have these zygotes, I can just take one zygote and see maybe the gender of that zygote and everything, because these Western guys, they want to see the gender and just specify the gender. Of course, haram Islam. And yeah, but we use PCR because just one cell, when you take just one cell, it's not enough. You need to amplify whatever you want to try to detect. So you use PCR for that. Okay. Now, this is the last thing. Khalas, alhamdulillah. Oh, we're already an hour. But believe me, just an hour, okay? I finished what Dr. Al-Jada took 3 or 4 hours. But it's really simple. Like, okay, it's a bit long because it's 3 lectures. It's not as easy as, for example, cell signaling, but it is not as hard as people make it to be. And this is now the last step, which is Sagner sequencing. Now we said about the sequencing, remember, and polyacrylamide and everything. So this is what we're talking about, sequencing DNA. How did we know, like, the human genome? How did they sequence the DNA? It's through a really, really, what I call, honestly, for me, it's like a genius way. I don't know how he thought about it, this guy Sagner, but for me, it's a really genius way. So what you'll do, okay, I'll have this fragment of DNA, okay, and I'll already have a radioactive labeled primer, okay. I know like a start of that fragment, and I'll have a primer attached to it, okay. And then what I'll do is I'll put it in 4 tubes. This is the old way. I'll put it in 4 tubes. Okay. I'll have the fragment of DNA and just amplify it and just have it in 4 single tubes. The same fragment in 4 tubes. Okay. For reaction mixtures. And then what I'll do, I'll add a dideoxy nucleotides. Dideoxy means that at, I think, the third carbon, there is no oxygen. So if there is no oxygen at that third carbon, okay, basically the PCR, when it takes that, when my polymerase chain, okay, where polymerase tries to amplify the DNA, and that takes that nucleotide and it puts it, and now it tries to add another nucleotide binding to it, it cannot. It just basically cannot, because there is no oxygen, so I cannot make that bond, that phosphodiester bond. So I'll just terminate it. And what I'll do, okay, so I'll add it to the DD adenosine, ATP, okay, and then one is for cytosine, dideoxycytosine, dideoxytyrosine, and dideoxyguanine, the 4 nucleotides that we use in DNA. And then basically, I'll just put it in a 1:10 ratio. I know it's a bit complicated, but just focus with me, okay? So I have this dideoxy, and it is
1/10 of the concentration of the normal nucleus, because I need to put normal nucleotides there so for the amplification to happen. Okay, but then whenever that dideoxy is taken by the polymerase, it will terminate the elongation, and I'll have just a fragment of the DNA. So let's say, for example, I have this, okay? I have this GCTTAS, okay? And now I add and have the primer and everything, okay? And now the first thing that will bind to the G is a C, right? So a C fragment, okay? A C, and then here it's gonna bind a G, and then here it's gonna bind an A will bind here, a G will bind here, a T will bind here. But for example, let's say if a C binds here, right? And let's say it's a dideoxycytosine. What will happen? I'll have this just a smaller fragment, 1, 2, 3, 4, just a 5 base fragment. So as you can see, it's the smallest fragment here. So this is my first base, okay? C. And then I'm gonna have, after Z, I'm gonna have a G, right? A G binds here. But for example, let's say that the dideoxy guanine was the one that binding here, so it will terminate it. So the second one will be the guanine, okay? And then the same goes on. And then T, okay, what will bind it is the adenosine. Let's say the dideoxyadenosine was added there. It will create just a 6 base fragment, and you can see it here, A. And I just move on, okay? I counted from the smallest. I start reading from the smallest fragment to the longest. And this was happened, okay? So C, the first one is C, okay? And then the second one is G. Third one is A. Fourth one is A. Fifth one is G, T, C, A, and then G. And because it's complementary, right? It's not my actual sequence. I'll just, through complementary base pairing, I'll read the original sequence. So I'll just say, okay, C binds with G. So the first one is G. Here is going to be C. A is going to be T. And then I just have read the sequence. Now, this is the old way where I put it in four reaction mixer tubes and everything. But now, what I'll do is for my dideoxy nucleotides, okay, I'll have them made out of different chemiluminescent materials. So, for example, my dideoxyadenosine will be red. My dideoxycytosine will be green. My dideoxythymine will be, I don't know, purple. And then guanine will be, I don't know, gray. Something like that, okay? And I can mix them together and just put them in the same sample and in the same concentration of 10% being dideoxy and 90% being the normal nucleotides. And then I just do gel electrophoresis, and then I can see the colors. So I just go like this. Okay, this is red, so the first one is A, and then the second one is gray, so it's guanine, and from like that, from just colors. And now I can just sequence the whole thing. And this signal sequencing and the gel electrophoresis is through the polyacrylamide. That's why we use for sequence, because just one base difference and can give you different bands. And this is signal sequencing. And yeah, for microarray, he was recorded, Dr. Ajada, and said, I know that I'm being recorded, and I will never ask you for microarray. If a question comes in the midterm, you can appeal it. You have me on record. But if you see a question, and for example, in the past or something when you're following past, it's mainly about cancer expression, something like that. So if you just want to solve it in the past. Questions, finally? Okay, so just read it. As you can see, this is, I took it from his lecture. He just wants you to think. This is just what actually never solved it. So yeah, this is the paternity. As I said, you just need to match the missing bands. That's from his, not from his mother. So you see, this band's from his mother, okay? But this band is not. Does his father have it? Yes, he has it. Okay, that's good. And then this band, okay, his father does not have it, but it's from his mother. So we don't care about it. And the last fragment, isn't it from his mother? No. But so does the father have it? Yes. So he fills the missing fragments, the missing band. So that means that this is the father. Correct maternity and paternity, because the mother will has the same the bands. Yeah. So basically, yeah, correct paternity and maternity. Yeah. So gene amplification is an increase of the number of copies of a gene without a proportional increase in other genes. This can result from duplication of a region of DNA that contains a gene through errors in DNA replication and repair machinery, as well as through fortuitous, I don't know how to read this, capture by selfish genetic elements. Which of the following techniques is used for the amplification of genes? Amplification, PCR. And now we will go to enzymology. But I think the longest is over. This is the longest quarter. The rest is gonna move easy. But as you saw, yeah, it's long, but it's not as hard as people make it. It's really not as hard. So now to enzymology. Okay, now enzyme and enzymology. We'll start with Dr. Ajeta's lecture. One, the introduction, and two, the one about the kinetics. So first, what are enzymes? Enzymes are biological catalysts. What does a catalyst mean? It means a substance that just speeds up the reaction. So enzymes are your biological catalyst that speeds up our biological reactions. And most of the enzymes are proteins, but sometimes they could be RNAs, as we have seen. They could be ribozymes or that type of RNA enzymes. So mostly proteins, but it can be also RNAs. And how does the enzyme speeds up the chemical reaction or speeds up our reaction? It basically just gives us an alternative pathway where there is a lower activation energy. What is the activation energy? It's the amount of energy needed to reach this state, which we call a transition state. And in this state, after becoming in the transition state, we will start to reach a state where the reaction moves on its own. خلاص. It will do the on its own. No need to pump energy. No more. So and this without an enzyme, it just pumps the energy, and then till it reaches the transition state. After that, it will just the reaction will move on its own. But with an enzyme, as you can see, the amount of energy pumped is less, right? It's just less energy to reach that transition state, and then you'll have your product. So this is basically what's an enzyme's effect. It lowers your activation energy. But as you can see, the energy difference between reactants and products is not changed with an enzyme or without. See, the energy difference is the same. Yes, maybe the activation energy is different, but the energy difference between reactant and products is the same. So that's something about we need to know. As you can see, reactant, the transition state, then your product. Yeah, and how does the enzyme actually do the reaction? Is basically takes the substrate, and it binds to a site in the enzyme called an active site. Okay, and from there the enzyme will make it favorable for the substrate to form the product, either like better collagen or something happens in the active site that favors the production of the product, speeding up the reaction and producing more products. There are two hypotheses of how an enzyme and a substrate interact, and the active site. One hypothesis is that the active site is a perfect fit for the substrate. So when a substrate comes, the active site is already a perfect fit for it, and then it will cause the reaction to happen, and then producing the products. Okay, converting the substrate into products. So that's the lock and key hypothesis. But there is another hypothesis, which is the induced fit. Okay, so basically, induced fit, that the active enzyme is in its form, right? But then when a substrate comes that is specific for the enzyme, what will happen? The active site will change depending on the substrate, and it will fit it perfect. Like at first, it's not fitting it perfectly, but when the substrate comes, the active site will change so it can fit the substrate. As you can see, this is, for example, hexokinase without C. How's the active site, but then when the glucose come, which is a substrate, it will change shape. The active site will change shape to fit it. It's basically like a glove, right? You take the glove, it does not perfectly match your hand, right? It's not. But then you put your hand inside the glove, and it fits your hand, and now it's look like your hand perfect. So it's an induced fit. Okay. And now there are three types of enzyme specificity. One is that absolute, so only one substrate and one substrate only. And that you can see for an enzyme urease. Urease is an enzyme that only catalyzes the hydrolysis of urea, so converting urea to ammonia and carbon dioxide. So only urea is its substrate. And where it's specific for a group of molecules, so for example, hexokinase, where it phosphorylates. You know that hexokinase job is phosphorylating sugars. So hexokinase is for all hexoses, all hexose sugar like fructose, glucose, galactose, all these hexoses. Uh, for hexokinase adds a phosphate group to them. And then you have a linkage where this enzyme is specific to one type of bond. So maybe a peptide bond, maybe some phosphodiester bond, some type of bonds. And it's just linked to that. And an example, as you remember in GIT, like carboxyaminoperoxidase or uh, amine aminopeptidase, basically, it just breaks a peptide bond. Whatever the protein is, even if it's two different proteins, it just breaks that bond and makes the single amino acid. I don't think the examples are needed, but just to make sure. And now we have 6 classes of enzymes. Unfortunately, I thought it was not important, but unfortunately, I saw some questions on it, so I'll just go on it fast, and I'll just give you some tips to immediately know which one is which. Oxidoreductases, redox reactions, is for redox reactions, and if you ever see like transfer of hydrogen and electrons, whenever you see electron carriers like NAD, FAD, that's an oxidoreductase. And then second class is transferases. It's the transfer of like methyl groups, phosphate groups, nitrogen groups. So when you see, like, for example, here there is a transfer of methyl group, but where you could maybe make a mistake is the transfer of phosphate groups. So when you see hexokinase phosphorylates glucose, right? You think that just adds a phosphate. No, it transfers a phosphate from where? From an ATP. So hexokinase is a transferase. So phosphorylation is basically a transfer of phosphate group from probably ATP into another molecule. So whenever you see phosphorylation, it's a transferase. And also, for example, you see the transfer of methyl groups. This was in B9 and B12, and how like you need to add a methyl group to folate and all that. But mainly, if you ever see phosphorylation, it's a transferase. And then you have hydrolase. It's basically when you break down a complex molecule into two simpler molecules using water. If water is a reactant, and then you see a complex molecule is turned into two simpler molecules, that's a hydrolase. As you can see, urea, well, kind of complex molecule, but then when it's added to water, okay, with urease, you'll which is our hydrolase, it will catalyze the breakdown of urea into carbon dioxide and ammonia. So that's a hydrolase. And the lyases is basically it sees a molecule and just breaks a certain bond. So if you ever see like one reactant and two products, as you can see, pyruvate, one reactant, and then two products, acetaldehyde, carbon dioxide, lyase. And then you have isomerase, it's just changing the molecular geometry of a substance. So if you see one reactant, one product, and there is no difference in the molecular weight, or just one reactant, one product, it's probably an isomerase. And then we have ligases. It's basically adding a carbon bond, sulfate, nitrogen, and just adding it together. So whenever you see two reactants, one product, that's a ligase. So let's go just fast. Electrocarrier. Electrocarrier, redox reactions, oxidoreductase. Transfer of carbon, or we see phosphorylation or anything like that, transferase. You see breakdown of complex molecules into simpler molecules, and water is a reactant, it's a hydrolase. When you see one reactant, two products, that's a lyase. When you see one reactant, one product, no change in molecular weight, that's an isomerase. When you see two reactants, one product, that's a ligase. Okay, now zymogens. Zymogens are basically inactive precursors of proteins, and they are turned into this active protein either by a biochemical change like low pH with trypsin, or maybe a cleavage of a peptide bond like that was making it inactive, like for example trypsinogen turned into trypsin. Yeah, so that's basically it. And now we have the temperature effect on enzymes. You see, the if when we go from like low temperatures, every increase of 10 degrees Celsius, we see that the enzyme activity, okay, increases by 2 to 3x. So from 10 to 20, the enzyme activity doubles or triples, and from 20 to 30, doubles or triples. And that's what we call the Q10 coefficient. And this is for all chemical reactions. But when it comes to biology and enzyme activity, we know that enzymes are most of the time they're proteins, even RNAs. They can be affected by high heat and the denature or breakdown. So enzymes will have this Q2 coefficient and the doubling and tripling of its activity until it reaches its optimum temperature. In humans, it's around 35 to 40. Some fish that lives in cold waters, their optimum temperature is at 30. After that, it's gonna be dead. And some bacteria that lives in very high temperatures, their enzymes optimum activity is at 70 degrees Celsius, like for example, Taq polymerase from PCR, and that's it. But the thing is, after the optimum temperature, if I increase the temperature, what will happen is the opposite of Q10, and the enzyme activity will decrease, as it will cause the denaturation of my enzyme. So after optimum temperature, there will be denaturation, and the Q10 coefficient will not be true anymore for the enzyme activity. And now we go to the pH effect on enzymes, and all different enzymes have different optimum pHs depending on the environment they work in. So, for example, pepsin, it works in the stomach, right? And the stomach is acidic, so its optimum pH value is at 2, an acidic environment. Trypsin, on the other hand, works mainly in the duodenum, which has an alkaline environment. So its optimum pH is going to be around 910, which is a basic pH. And for pH changes, a simple pH change could cause a drastic change in the enzyme activity, as you can see, from 3. And then if I just make it to 5, see how's it? Almost halves. And the same thing for trypsin. So pH changes can affect the enzyme activity drastically. And now we have ion effect on enzymes. Just know that like salt concentration, ion concentrations like sodium, chloride, potassium, calcium, they affect the enzyme activity. So we need optimum salt ion concentration for optimum enzyme activity. Now for enzymes, coenzymes, and cofactors, basically, if an enzyme needs a cofactor or something to be activated, before it binds to that coenzyme, it's called an apoenzyme, just like a portion of the enzyme. It's not whole, it's not complete. But then when it binds to its coenzyme, activating it, it's going to be called a holoenzyme, a whole enzyme. And that's where it can be activated. And there's different types of coenzymes and cofactors. Like, for example, you have the normal coenzyme, which is an organic molecule and probably derived from vitamins. As we can see, B6 is a very common coenzyme. We can see it as a coenzyme for many reactions. It's not as I'm just giving you an example. And if the coenzyme is and coenzyme, as you can see, is loosely bound to the enzyme. It's not tightly bound. And if that coenzyme is altered after the reaction, it's called a co-substrate. And an example of that is NAD. We'll talk a bit more after this slide. And now we have a prosthetic group. It's a coenzyme, but it's tightly bound to the enzyme, okay? And it's immediately turned to its original form. And it's tightly bound. It's there all the time. And then we have apoenzyme and a cofactor. Sometimes when we say cofactor, it's probably an inorganic, probably like iron or copper. Some enzymes need some types of metals, ions, to be activated. We can see coenzymes, the difference between co-substrate and a prosthetic group. The co-substrate is loosely bound, it dissociates in altered state, and it's always recycled. An example of that is NAD, with, for example, ketoglyceride dehydrogenase, isocitrate dehydrogenase. NAD is a co-substrate. But for example, when you go to a prosthetic group, for example, succinate dehydrogenase, these are all examples. You don't need to memorize. I'm just giving you, if there is something that you need to memorize, I'll probably put it in the slide. Don't worry. A prosthetic group is tightly bound, Okay, so for example, you know that succinate dehydrogenase, the TCA cycle, it's only found at the inner membrane of the mitochondria. Why is that? Because that's where FAD, the prosthetic group that it's tightly bound to, it's where it transfers its electron in the electron transport chain. So whenever a succinate dehydrogenase gives the electrons to FAD and it reduces it, FAD will immediately be oxidized, okay, giving it its electrons to the electron transport chain, and will return to its original form and tightly bound to that enzyme, which is succinate dehydrogenase. So that's basically it. And now we have enzyme units. We have Qatar, which is not used a lot. It's basically how like the amount of product an enzyme can produce in mole units per second. But mole is 1 mole of like glucose is very large, like 300 or 500 grams. I don't know, but mole is a very large quantity. It's not applicable to us humans. So what we did is we make it in the international and enzyme unit, U or IU, which is millimole. Milli is 1 millionth, micromole. I mean micromole, which is 1 millionth of a mole per minute. So how much micromoles of a product an enzyme can produce per minute? And that's our enzyme unit activity unit that we use most of the time. So when you see 5 units of insulin, means that this amount of enzyme uh, can produce 5 millimoles per minute of the product. And now we go to enzyme kinetics. As you can see, you just need to know that on all these graphs, our enzyme activity, our enzyme concentration is fixed. It does not change. So when we have this rule, I can explain everything much easier. So now that, for example, you can see as substrate concentration increase, the velocity and the reaction velocity and the amount of product that is produced increases, normal, okay? But then it reaches a plateau. At this type of concentration, now it reaches a plateau. It cannot increase. That's the maximum velocity. Why? Because at that amount of concentration, all the active sites of the enzyme are filled. So the reaction cannot be any faster. Already all the active sites are filled. So now that's where I reach what I call the Vmax, the maximum velocity. And as you can see, this is a hyperbolic curve, okay? And it's from the Michaelis-Menten equation and model. I'll explain it later at the end of kinetics. But basically, what you need to know is the Michaelis-Menten constant, or Km. It's basically just means the substrate concentration where Vmax or half of the Vmax is reached. Basically, it's basically that. And this mainly tells you about the affinity. So the higher the Km, okay, that means the higher substrate concentration is needed for reaching just the half of the Vmax, which means there is less affinity, because I mean I need to have higher concentration of the substrate just to reach half of the Vmax. But when it's a lower Km, means I just need a lower concentration of the substrate to reach half of the Vmax, which means that the affinity is higher. So Km and affinity are inversely proportional. The higher the Km, the lower the affinity. The higher the affinity, the lower the Km. Basically it. As you can see, the Km has very big importance. For example, glucokinase. As you know, glucokinase, normal situations, it will not be working because its Km is very high. So at normal glucose concentrations, which is probably 0 to 5 millimoles per liter, only hexokinase work because it has a very low Km. Its affinity is high to glucose, and it works in uh, normal blood glucose concentrations. But for example, after you eat a large meal, right, and you don't want your blood glucose to stay very high, so you need now an enzyme that has a higher Vmax, okay, and that doesn't work in normal glucose concentration, because I don't want to have hypoglycemia where it takes all the glucose. So that's where glucokinase comes. It only works when there is high blood glucose concentrations, and it has a very high Vmax, so it can maintain immediately the normal limit of blood glucose. And we can see it also in the alcohol metabolism. If you know Asians, right, they cannot be drunk. They have something called the Asian flush. Like immediately after they drink one sip of alcohol or like a bit of alcohol, they immediately get all this allergic reactions and get hangover. Why? Because there is a high accumulation of the acetaldehyde, okay, CH3CHO. So how's that? Why do they have higher accumulation of acetaldehyde per drink more than normal other ethnicities? Because their alcohol dehydrogenase, which turn alcohol immediately to acetaldehyde, has a much lower Km, so it has higher affinity, so it immediately turns alcohol to acetaldehyde. But then their aldehyde dehydrogenase variant, it has a much higher Km, so much lower affinity. So acetaldehyde is produced quickly, but then it stays there for a long time until it's converted to acetate, the normal form. So that's why Asians hardly can get drunk. They get hangover very fast. And now we have allosteric enzymes. Now we know that in every chemical pathway, I have my committed step, which means if that step is happened, and now I convert this product into B, خلاص. Now the pathway will move on, and I'll get my end product, which is F. And we know what is the committed step in, for example, glycolysis. If you remember, it's step 3, and it is turning fructose 6-phosphate into fructose 1,6 bisphosphate. And usually these steps, okay, these committed steps are the most regulated steps. Like it has the most activators and inhibitors, okay? So it cannot be activated immediately. So when you see them in the graph and when we increase the amount of substrate, it does not immediately start to increase in the hyperbolic curve. No, it has more of a sigmoidal, more of an S. Why is that? It's basically because at first you cannot activate it immediately. It needs a bit more, okay? But then when it reaches that threshold, okay, it will start now increasing until it reaches its plateau, the Vmax. So that's basically how committed step, how the committed step and enzyme activity at that committed step, that's how it works. It has a sigmoid curve. And the enzymes that catalyze these committed steps, it's called allosteric enzymes because there is lots of allosteric regulation of that enzyme. So, for example, I have ATP and citrate inhibiting phosphofructokinase, which is my allosteric enzyme here. And this inhibition happens through allosteric regulation. And then I have ADP and AMP. They also regulate this phosphofructokinase, activating it. So you can see there is lots of molecules that regulate this enzyme allosterically. Some will be activators and some will be inhibitors. And it's always this sigmoid curve is always found in your committed step. But other steps, the normal step like E2, E3, these steps, they will show the normal Michaelis-Menten graph and model. But committed step and allosteric enzymes, they will show the sigmoid curve. And now we talked about activators and inhibitors, right? Now enzymes have two states. They either have tight state, T state, okay, or they have R state, relaxed state. So basically, and normally, inhibitors, they want to remain the T state, the tight state. It's tight. It will not do any. It will not do any reaction. Okay, but then activators, right? They want the relaxed enzyme that will take all the substrate and convert it into a product. So that's basically it. R state relaxed, T state tight. Inhibitors will go with the T state. Activators will want the R state. And of course, the T state will have low affinity. The R state, the relaxed state, will have high affinity. And all these activation and inhibition is through allosteric sites. Allosteric sites is sites different than the active site, which regulate the enzyme activity. And from here, with this Michaelis-Menten graph, okay, what I want you to see, this is, as you can see, this is what a sigmoid curve. That means it's a commit step, okay? It's not a normal hyperbolic. And with this, and whenever you see this graph, especially this graph, okay, this hyperbolic graph, whenever the graph has a left shift, okay, see, this is the normal one, right? Without anything. If it goes to the left, that means there is an activation. If it goes to the right, that means there is inhibition. Basically that. And now we go to like clinical, like allosteric enzymes, what the importance of regulation. We have Gout's disease, which is basically when the purine synthesis, like the synthesis of adenine and guanine for my DNA or even RNA, right? We need to synthesize them, right? But we need to synthesize them in a regulated manner. I don't want too much, I don't want too little. But with God's disease, there is a loss of regulation of the synthesis. So I'll have overproduction of purines, I'll have overproduction, and that will cause gouts where there's accumulation of uric acids in joint. Just know that gout is due to loss of allosteric regulation of purine synthesis. That's it. Now, we need to talk about inhibitors. We have two main types of inhibitors. We have first the irreversible inhibitor. That means when if it binds to the enzyme, it will inhibit it forever. And then we have the reversible ones, which are three types: competitive, non-competitive, and uncompetitive. The competitive one is the one that we use mostly in clinical situations. But let's start with the reversible. That's where we go to poisons. Okay. For example, pesticides, they have this compound called organophosphates. Okay. And these organophosphates, what they do, they're irreversibly bind to an enzyme called cholinesterase. And when you block this enzyme, you'll basically block acetylcholine transmission in neurons. And that will cause paralysis and very severe effects and eventually death. And this is actually the organophosphates is what we use for like assassination poisons. For example, this is just an example for me. It's a fun fact. Russian intelligence, they frequently use Novichok agents, which are fourth generation organophosphates. And they use it mostly for, as I said, assassination. And of course, because they're organophosphates, they target acetylcholinesterase. Now we go to the Michaelis-Menten model. Michaelis-Menten model will produce you this hyperbolic curve, and it's based upon this like reaction. Basically, first I have the enzyme plus substrate, okay, and then they transform to the transition state where there is the substrate bound to the enzyme, and then second thing happen, and then I have the enzyme, the active enzyme plus product. So now it dissociates and the product is produced, and produces you this reaction, okay. You do not need to know the reaction. There were hardly any questions about the reaction, but I need to know, to explain for you the reciprocal of that reaction. So when I just reciprocal is basically I put 1 over everything, 1 over V, 1 over V max, 1 over all of this, and it will produce this reaction. 1 over V equal Km over V max, 1 over S plus 1 over V max. I know it looks complicated, but it's actually just Y equal Mx plus C. Okay, you know, just the straight line that you have learned in 6th grade pre-algebra. This is basically it, where 1 over V is your Y, so it's your Y variable, okay? It's your vertical one in the graph, right? The vertical one in the graph. And then I have Km over V max, which is my M, and M we know is the slope. So Km over V max is gonna be the slope of that equation, okay? And then X, okay, is 1 over S. Okay, so the horizontal line, right? That's gonna be 1 over S. And then C, which is my y-intercept, it's gonna be 1 over V max. So why do we need to learn this? We get a straight line, okay? So for example, you want to find V max, right? I said that C in that equation is 1 over V max. So the y-intercept is 1 over V max. So let's, for example, say that the y-intercept is 5, right? What's my V max? I just change it. One is going to be, if it's 5, the intercept is 5. That means V max is 0.5, right? For example, what if the intercept is 0.1? 1 divided by 0.1. So my Vmax is gonna be 10. And that's basically it. That is what is Vmax. So, and now I want to find Km. I've already found Kmax. So what I'll do, I just find the slope of this line, and then it's gonna be that slope equal Km over Vmax. I can find Vmax from the y-intercept, and then just Vmax times the slope. It will give me the Km, or even the x-intercept. It's equal minus 1 over Km, and that will tell me what is Km. So if like Km was minus 1, right? That means my Km is 1, which is my Michaelis-Menten constant. And yeah, but the most important thing I just needed to know is that the slope is Km over Vmax and the y-intercept is 1 over Vmax. Basically that. Just know that for now. So now we go to the inhibitors. We have first competitive inhibitors. And what they do is they just basically compete for the active site, right? Compete for the active site. And whenever they bind to the active site, the substrate cannot bind to that active site, which will lead to the no reaction has no product. So what does competitive inhibition do? Okay, it decreases affinity, so it increases Km. Okay, but the Vmax is not changed. Why? Because if I increase the substrate concentration, right, and now it's way more than the competitive inhibitor, the reaction will just move slowly and then I'll reach the normal Vmax. Okay, it will overcome the inhibition easily. But because there is a competition, right, there will be less affinity, so the Km is going to be higher. But Vmax is not changed because the amount of active enzyme is not changed. Okay, the enzyme is still not there. There is no allosteric regulation in like inactivating the enzyme. No, it's just the competitive inhibitor just preventing the substrate from binding. So with competitive inhibition, Vmax stays the same, but affinity is lower, so my Km is higher. And an example of a competitive inhibitor, which is most of the drugs that you will prescribe as a doctor, I know Al-Jada says it 100 times, is a competitive inhibitor. And the example that you need to know is statins, the one that, for cholesterol and that. So the statins are an example of a competitive inhibitor, and they inhibit the enzyme HMG-CoA reductase, which is the enzyme for the committed step in cholesterol synthesis. And now we have non-competitive inhibitor. Non-competitive inhibitor is just, it has an allosteric site, right? As an allosteric site. And when it ever binds to that allosteric site, it inactivates the enzyme. Whether the enzyme is with the substrate or whether it is without the substrate. It just, whenever it binds to that allosteric site of the enzyme, doesn't matter if the active site is filled or not, it will inhibit it. And that means it lowers the amount of active enzyme. So what can we say? We can say that it lowers Vmax because the amount of active enzyme is now lower. But does it affect affinity? No, because it does not compete for that active site. It does not affect that active site at all. Okay? So affinity is not changed, so Km is not changed, but my Vmax will be changed. So non-competitive inhibitors lower Vmax, Km stays the same. Uncompetitive inhibitor, well, basically what it does, it inhibits the enzyme substrate complex. So whenever, so let's say that it binds to an allosteric site, okay, but it does not bind to the enzyme when it's alone. It only binds to that allosteric site when the enzyme substrate complex is formed. So first the enzyme binds to the substrate, and when it's khalas, I'm gonna produce the product, the uncompetitive inhibitor comes and then just freezes it in the enzyme substrate complex. So we can see that it lowers the amount of active enzymes because it binds to an allosteric site, so inactivating the enzyme. So amount of active enzyme is lower. So now we would say that it lowers Vmax, but it also increases affinity. It decreases Km, increases affinity. Why? Because basically what it does, it will lower the amount of products, okay, and it will try to shift the reaction into E plus S. But then we have something called Le Chatelier principle, which basically tries to restore equilibrium. Okay, so if something tries to make something go the other way, it will counteract it, okay, by trying to maintain the equilibrium. So what's the counteract? It's by increasing affinity, because it's like there is less enzymes now, active enzymes. So whenever it finds a substrate, it will be very high affinity. So just know that uncompetitive inhibitors, they lower both. They lower Vmax and Km. And lower Km means increased affinity. And now just the identification points, if you ever have it in a graph. Whenever you have equal y-intercept with inhibitor and normal activity, that's a competitive inhibitor, because If it's equal y-intercept, that means it's equal Vmax. And the only one that does not decrease Vmax is competitive inhibitor. And whenever you see like parallel lines, like same slope and like parallel lines, there is nothing different, that's an uncompetitive inhibitor. Okay, because they decrease Km and Vmax equally, so the slope will stay the same. So whenever you see parallel lines and equal slopes, that's uncompetitive inhibitors. And if you see no equal Vmax and no parallel lines, just very random, that's a non-competitive inhibitor. That's it. And now for questions. Kinases are a class of enzymes that incorporate a phosphate into the substrate. The catalytic activity of kinases classifies them as members of which of the following enzyme families. Incorporate a phosphate. You know, he tries to make it hard, but incorporates a phosphate. We said phosphates are what? They're transferases. And now for the Lineweaver-Burke plot is basically the reciprocal equation, right? The red line was mostly likely obtained by addition of which of the following? So it's an inhibitor, okay. But which inhibitor? I see an equal y-intercept. So that means it is a competitive inhibitor. And now we go to clinical enzymology. We start first by there is functional enzymes in plasma and non-functional enzymes in plasma. Whenever we detect like enzyme presence in plasma, that we have functional enzymes and we have non-functional enzymes. Functional enzymes are like your clotting factors, your ceruloplasmin, for blood clotting, something like that. Usually, they have a specific function in plasma, as we know. Their concentration are large, and it's a big problem if they have low concentration. But then we have non-functional enzymes, enzymes that that has no relation to be in the plasma. They shouldn't be in the plasma, so they have no specific function in the plasma. Most of the time, and their plasma concentration are very low because they don't have any function. Why would they be there? And it's very bad if their concentration are high. Okay, so how do non-functional enzymes come to the plasma and have high concentration? Sometimes when you have tissue destruction like cirrhosis or myocardial infarction, the enzymes that were found in the heart or the liver, they will leak to the blood, which says, oh, there was a problem in the heart, oh, there was a problem in the liver. So yeah, basically, and examples of my non-functional enzymes are like LDH, CPK, alpha phosphatase, aspartate amino acid. Don't worry, we will talk about them all. Okay. So the tissue damage, maybe heart tissue or liver tissue or even muscle tissue, will lead to the leaking of these enzymes into the plasma. Now, when we talk enzymes in disease, maybe the deficiency of that enzyme is the cause of the disease. For example, you remember when we talked about carbohydrate metabolism and all that, and maybe when we have fructokinase deficiency or galactokinase, or these enzymes, when they're deficient, I'm gonna have accumulation of some metabolites which are toxic sometimes in the body. So, for example, when we have fructokinase deficiency, I have accumulation of sorbitol, which will cause oxidative damage. I have cataracts, maybe I'll get blind. There will be sperm infertility and all these things. But then it also can be a congenital disease like Pompey's disease, the absence of the 1,4-glucose, like you cannot break glycogen. I forgot the name of that enzyme. But sometimes the abnormal enzyme activity or abnormal amount of enzyme is just a secondary effect of a greater problem. So when I have, for example, a myocardial infarction, I'm gonna see abnormal amount of cardiac enzymes in my blood, which indicate to me it's not the amount of enzymes or the presence of these enzymes in the blood is not the disease, but they indicate, oh, this guy might have had a heart attack. So that's basically it. And this is the enzyme profile in disease. First, we have so these you need to memorize. Okay, unfortunately, you need to memorize because they can ask you a question where, oh, this enzyme was elevated. So, what disease did this guy have? Which organ? So you need to memorize them. In myocardial infarction, we have CPK2, which is creatine kinase variation 2 with MB. Okay, just it has the MB factors. We'll talk about it, but just know CPK2. Okay, and then we have alanine aspartate transferase AST. Okay, and we have LDH1 H4. Okay, so the first variant of LDH, because there are 5 variants of LDH, by the way. So it's LDH1, it's a myocardial infarction. It's related to the heart. And then another one, which is the most important one, troponin. It's troponin I, troponin T. Whenever you see troponin, elevation troponin, that's probably about the heart. And then for liver disease, we have a lot. We have ALT and AST, but mainly ALT. AST is related more to the heart and muscles, but ALT is mainly for the liver. We have alkaline phosphatase. It's seen in liver and like alcoholic and hepatitis, but it's also found in bones. Okay, so if you see ALP and there is bones in liver, most probably choose bones. Okay, but then these two are mainly liver. Why guatematin peptidase, something like that? Liver immediately. ALT, liver immediately. And LDH5 M4, this is found mainly in liver disease, sometimes also in muscle injury. And with muscle disorders, yes, as you can see, LDH5, and we have creatine kinase, the third variant, which is This is related only to muscles, and as you can see here, AST. But mainly for muscles, CK3 is the golden one for muscles. Also, myoglobin is found in muscles, because we'll talk about myoglobin heart, but it's also found in muscles. And as we said, with bone diseases, it's mainly alkaline phosphatase. If you see alkaline phosphatase and bone disease in the choices, choose always alkaline phosphatase. And then for pancreatic diseases, you have alpha amylase and lipase. But lipase is way more specific for pancreas, and it's the most common. Alpha amylase is, if you see lipase and alpha amylase for pancreatic disease, always choose lipase, because alpha amylase can be seen in where? In salivary glands. But lipase is specific for the pancreas. And then we have, I don't know why it's not showing, it's not loading. It's basically problem here. I think because I'm offline. Well, it's basically the cereoplasmin. Okay, it's an enzyme found in uh, it's found in plasma. It's for oxidizing or reducing iron. Just know cerebroplasmin is a plasma enzyme, and it's a functional enzyme. And if it's slow, that's a problem. And now we have creatine kinase. There are like isozymes, right? So there are different variants of an enzyme that does the same thing. And for creatine kinase, it has 3 variants. Okay, it has, when it's BB subunit, because it has 2 subunits, M subunit and B subunit. If the composition of the subunits of creatine kinase is BB, brain. If it's MM, muscle. And if it's between them, muscle and brain, MB, heart. So if there is a release of CK1, probably a brain or CNS disease. If there is a release of CK2, creatine kinase 2, it's probably a problem in the heart. And with CK3, which is MM, it's probably a problem in the muscle. Now, when we go to LDH isozymes, okay, as we said, there are 5 variants. I just need to focus on LDH1 and LDH5. If there is a release of LDH1, probably something with the heart, myocardial infarction, okay. And if it's a release of LDH5, problem with muscle damage, okay, or cirrhosis, hepatitis, muscle problem with the liver. And for LDH1 measurement, so basically for heart attacks, this was before when we used to use LDH1. Basically, you just need to know that LDH2 should be higher than LDH1. But if we measure and we see that LDH1 is higher than LDH2, there is a shift. That means that we might have a myocardial infarction. And, enzyme release after myocardial infarction has a lag phase, right? Every enzyme has a lag phase. It will not release immediately and we can detect the plasma immediately. No. Some enzymes will take 2 hours for them to start appear in the plasma and the blood and have a high level. Some, like the lactate dehydrogenase, takes 1 day. So you have the myocardial infarction, all that pain, but lactate dehydrogenase, which is an indicator of your heart attack, will only be increased in the plasma after 1 day and peaks around 2 to 3 days after your myocardial infarction, and to stay elevated till 5 to 10 days. And that's basically it. And for us, for the ideal cardiac marker, like the best cardiac marker we can find is a marker that's only specific to the heart. We don't find it anywhere else. So if it's elevated, we know that's from the heart. Okay, it releases immediately. It does not have like a long lag phase, right? It releases and start to be elevated the blood a short time after the problem happened, after the myocardial infarction, and that it's gonna be easy to perform, and that also has a long half-life, so it stays a lot. So if we want to detect it, if there is like a late detection, we can detect, oh, you had a heart attack, even though it's 5 days probably after you had that heart attack. And now we have old versus new cardiac markers. Before, we used to have total CK, right? So the CK for the brain and the muscle and the heart, we used to measure it with LDH1, the one we just talked about, and AST. But you see, AST and CK, total CK, are not specific for the heart. And the problem with LDH1 is that it takes 1 day for it to be elevated. So 1 day after the heart attack. So these are the old cardiac marker. For us, there are new cardiac markers, the CKMB, the creatine kinase specific to the heart, myoglobin. It's not used a lot, but just the start one. If we see it, it does not mean there's a heart attack, but maybe he has a heart attack. Let's wait for what? For our golden boy, troponin. And if troponin is elevated, that means probably heart attack. And so like the golden marker. As you can see, these are like when they start to get elevated and when they are in their peak and when they return to normal levels. As you can see for myoglobin, it's like 2 hours of the heart attack, it will peak, but then like 20 hours after, khalas, it will return to normal levels. So it has short half-life. CKMB, as you can see, it like start to rise like after 4 hours, 4 to 8 hours, and then peaks around 12 to 24 hours after the myocardial infarction, but then it returns to normal level after around 3 days. But the troponin, which is the golden marker for cardiac diseases, it takes around 48 hours to be elevated, okay? But then it peaks around 12 to 16 hours, but then it stays elevated for around 6 to 10 days. So very long half-life. So we can have like late detection. So it gives you a lot of time to detect, oh, you actually had that heart attack. And if they ask you about lag phase for myocardial infarction and that, you cannot say myoglobin because myoglobin does not determine, oh, he has a heart attack. It said maybe has a heart attack, but not definitely. But when we go to troponin or CKMB, because they only elevate after 4 hours, so that means the lag phase between myocardial infarction and determining if he actually had it is 4 hours. And now we have therapeutic enzymes. We have, for example, because enzymes are not just used for laboratory and detecting diseases, we can use it to treat disease. And for example, if we have like a blood clot or maybe a heart attack due to a blood clot, ischemia, something, we can use the enzymes tissue plasminogens, streptokinase, urokinase, same name for the same enzyme, which basically breaks these clots and resolve your heart attack. And for there are other things like asparaginase, okay, which is for leukemia, and then we have trypsin and lipase. If we have pancreatic insufficiency, we can inject these for to treat these problems and have these enzymes fill the place. So they are therapeutic enzymes, okay. And then at the end, we have laboratory enzymes, the ones that we use to diagnose disease. These are actually called clinical enzymes, okay? I think clinical enzymes. So just when you see them elevated in blood, okay, there's a problem there. But laboratory enzymes is the one that we use like in laboratories, right? For example, your tag polymerase, your antibodies that we use in ELISA, I still say ELISA, and all these are your laboratory enzymes, okay, that we use for specific molecular techniques. So these are your laboratory enzymes. And now we go to questions. First question: 57-year-old man has returned from overseas trip and he had severe substernal chest pain 3 days ago. So maybe a heart attack. Which of the following is most appropriate laboratory test to order for this patient? Hmm. Who's the golden boy? It's troponin. As we said, troponin, because 3 days ago, you cannot really use creatine kinase because after 3 days, it's probably in normal levels. But troponin stays elevated for 4 to 10 days, 6 to 10 days. So if it's late detection, it's mainly troponin. It's always troponin, actually, for myocardial infarctions. Which biomarker is most commonly used to diagnose pancreatitis? So an enzyme that's specific for pancreatic problems. Lipase or alpha amylase? It's lipase. And yeah, we are done with enzymology. Now we'll go to the metabolism, insulin, glucagon, feed fast cycle, and diabetes. It's gonna be easy, inshallah. Okay, now moving on to the metabolic effects of insulin and glucagon. We'll just discuss the insulin, its structure, stimulation, inhibition, and its metabolic effects, while doing the same for glucagon. It's gonna be super quick, and it's not gonna take that much time. So we start now for the we are we're on the last quarter of the crash course, and this is mainly about fuel metabolism, about glucose, fat, proteins, how they metabolize. So, and the four major organs that we're gonna see recurrent and when we're talking is the liver, adipose tissue, muscle, and the brain. But for now, we're focusing on glucose. So when we're talking about glucose regulation, it's always the liver that's the main organ in glucose regulation. So now let's start with the insulin structure and production. First, as we all know, insulin is produced from beta cells that are found in the islets of Langerhans inside endocrine pancreas. Okay, so B cells, they produce this insulin. What is the structure of this insulin? Well, the active structure of insulin is two chains. Okay, it's 51 amino acids, 30 in the B chain, 21 in the alpha chain. I don't think you need to know the amount of amino acids in every chain, but what you need to know that there is two intercellular chains, intercellular disulfide bonds. See, these are disulfide bonds that are interchain. Okay, so between the two chains, there is two disulfide bonds. So two disulfide bonds that are interchained. Okay, but there is one intra-chain that inside one chain, which is inside the alpha chain, we have one disulfide bond inside the alpha chain. So just think intra-chain, intra-alpha. So one disulfide bond when it's enter between alpha and beta, two disulfide bonds. And insulin is an anabolic hormone. Okay, so it promotes glycogen synthesis, it promotes lipid synthesis, it promotes protein synthesis. So it just promotes the synthesis of everything. It just wants to build up. It does not break down anything. And basically, it's biosynthesis. As we said, that was the active form, the alpha and beta chain. But before it's active, what we call pre-proinsulin. Okay, then we cut out signal sequence. Don't need to worry about that. But then we have this is the beta chain, this is the alpha chain, and this is then the C peptide. And now when I want khalas to release the insulin, what I'll do, I'll just cut the C peptide and then immediately send the insulin to my bloodstream. But the C peptide is also sent to my bloodstream, and we can use it as a marker for insulin synthesis and insulin release. Why? Because first of all, C peptide has a much longer half-life than insulin in the blood. Insulin stays in the blood for like 6 minutes. You almost cannot detect, but C peptide has a much longer half-life, so I can detect it. And C peptide is released in equal concentrations. For every one insulin produced, there is one C peptide produced because they're released together, right? Now, C peptide does not have any specific function, but it has detection function. And we also can use it maybe if a patient is diabetic and they have like exogenous insulin, take it to them. That exogenous insulin does not have a C peptide. But I want to measure, okay, how good is this pancreas? How much insulin is it producing? And then if I try to measure the insulin, I'll measure the exogenous and endogenous. So the one produced by the pancreas and the one he's injecting. I cannot differentiate. How do I differentiate? I can measure the C peptide, which is only found from the insulin produced from your pancreas. Okay, so it has longer half-life, it is produced in equal concentrations, and it's only produced with endogenous insulin, the insulin that is produced from your pancreas. Okay, and now we go to insulin regulation. First, what stimulates insulin? First of all is, of course, as we all know, high blood glucose. When after you eat a meal or anything, you will have high blood glucose, which will stimulate the B cells. Because why? B cells have what we call GLUT2 channels. Okay, GLUT2 channels are only work at high glucose concentrations. So not a lot of glucose goes into these channels if it's in low concentrations, but if it's in high concentrations, what will happen? A lot of glucose will now go through these channels. Okay, and now this glucose, as you can see, GLUT2 inside the B cell, right? And then now it will undergo glycolysis. Okay, it will undergo glycolysis and the citric acid cycle and then phosphorylation. But where is the insulin production? Okay, you told us it does all these things, but where is the insulin stimulus? Where now, okay, yalla, I'll release insulin. Now, from all these glycolysis, citric acid cycle, oxidative phosphorylation, I'll have what? A very high surge of ATP. I'm going to have a high level of ATP. What will this ATP do? It will go to this potassium channels in the B cells, and it will basically block the potassium efflux. So potassium going out, it will block it. So the potassium will stay. Okay, and that will cause depolarization of the cell. Okay, there is no potassium efflux. Okay, and now this is negative. This outside of the cell is becoming negative depolarization, right? And now what will happen is what? I'll have now high calcium influx. Why? Because now I prevented the potassium from coming out. Okay, there is no equal positive charge. There's no equal charges now. I'm gonna have depolarization, and then I'll have calcium influx. So now I have first glycolysis, citric acid cycle, high oxidative phosphorylation. I'm gonna have high ATP. ATP will block potassium efflux, and this potassium channel, ATP gated potassium channel, this will cause depolarization, and now I'll have calcium influx into the cell. And this calcium influx will cause the insulin granules inside the cell to be released outside, and there you have insulin secretion. So first I have glucose coming in through GLUT2, and then I have glycolysis, oxidative phosphorylation, citric acid. I'll have high ATP. ATP will prevent the potassium channel. I'll have then depolarization of the cell, where I have calcium influx into the cell. The calcium influx will cause insulin secretion. Now, this is basically how insulin is secreted, and sometimes with diabetics, maybe if they don't have that much insulin secretion, what I'll do, I'll give them a drug called sulfonylureas. Okay, sulfonylureas. Let's see, what is it? It's called here. Sulfonylureas. Yep, sulfonylureas. Basically, they will, rather than just ATP blocking it, they will do it themselves, and then they will block this channel themselves. That will cause the calcium influx, and then I'll have insulin secretion. Okay, what inhibits insulin? Basically, you have your epinephrine, your catecholamines, epinephrine, norepinephrine, which are released basically in physiological stress. When you need high blood glucose, you need glucose to go throughout your blood to get energy, so your muscles can get energy, your brain can get energy. So this is during stress situations, epinephrine, norepinephrine goes to the bloodstream, stops insulin secretion because I need high blood glucose. I don't want my blood glucose to go down, and then I'll have all these metabolic effects. And of course, when you eat, you'll have insulin secretion, but through what? Not just by the high blood glucose, but also when you eat, there are some hormones are called glucagon-like protein or gastric inhibitory polypeptide. Glucagon-like protein is like, the glucose, insulin dependent, I don't know that long name we studied in GIT, it's the same, okay? The gastric inhibitory polypeptide is the same one as that, the glucose insulin polypeptide. So these, when you eat, there will be your GIT will secrete these hormones, okay, and that will cause my insulin stimulation and insulin secretion. And actually, this is why if I inject glucose, okay, intravenously, okay, so I'm gonna put glucose intravenously, and I'll just put glucose here inside the veins. And when I eat glucose, which one do you think will have higher insulin secretion? It's actually my orally ingested glucose. Why? Because when I put glucose intravenously, the only thing that's going to stimulate my insulin is just the high blood glucose, and that takes a bit much of time for the glucose to reach the pancreas and then release the high insulin, right? It's going to take time. But when I eat the orally ingested sugar, the glucose, what will happen? I'll first have the glucose going through my GI tract, right? And then I'm going to have these hormones released, And with these hormones, I'm going to have now the high blood sugar. These two in combination, they will call what we have the incretin effect, when insulin secretion is increased from orally ingested more than intravenously. And as we said, not only glucose, like in meals overall, when you eat, they will increase these peptides, so amino acids, fatty acids, gastrointestinal, they all increase insulin secretion. Now, how does insulin affect carbohydrates? Well, in insulin, we have, for example, in the liver, it decreases glycogenolysis, so the breakdown of glycogen, it decreases it. It decreases gluconeogenesis, the production of glucose. Again and again, we already have high blood glucose, so it will inhibit it. Okay, but then it will increase glycogen like the glycogen synthesis. I don't know the specific name. Glycogen synthesis in liver and also in muscle. Okay, and it will also cause the increased uptake in GLUT4 transporters. Where do we find GLUT4? In muscle and adipose tissue. Insulin only increases glucose uptake in liver and muscle and adipose tissue because they have GLUT4. Because GLUT4 is the only glucose transporter channel that is insulin dependent. Okay, it's the only one, and you only find it in muscle and adipose tissue. And insulin, when it's secreted, it will increase the glucose intake by these two things. And lipids, as we said, insulin will increase the synthesis. Okay, the synthesis. So what will insulin do? It will increase, it will first deactivate what we call hormone sensitive lipase. What does lipase do? It basically breaks down my adipose tissue. But I want synthesis, right? So I'm gonna inhibit it. Stop. HSL, hormone sensitive lipase, you need to stop breaking lipids. I'm gonna inhibit you. Okay? But then what will insulin do? Because it increases glycolysis and the glycogen synthesis and these things, right? I'm gonna have increased glycerol 3-phosphate, which is like one of the molecules that you find in glycolysis pathway. Glycerol 3-phosphate will increase, and this then can be used for what? It will reach now the lipids, and this glycerol 3-phosphate can be used for triacylglycerol synthesis. So basically, triglycerides synthesis in your adipose tissue. And this triacylglycerol increased with already my fatty acid, it will come together and it will form adipose tissue or triglycerides. But from where do I get my fatty acids? Well, with increased insulin secretion, I'm gonna have, of course, with increased food, right? I'm gonna have increased VLDLs and chylomicrons. Basically, these are the forms where we find lipids in blood. And then insulin, what it does, it will increase a protein called lipoprotein lipase. So it inhibits hormone-sensitive lipase, which breaks down adipose tissue, but it activates another enzyme called lipoprotein lipase, which breaks down my VLDLs and chylomicrons, so my blood fats. Just think of them as your blood fats. It will and lipoprotein lipase will break these chylomicrons, these VLDLs, and then I'll have a lot of fatty acids, release of fatty acid into my adipose tissue. Now I want to the triglycerides. I want adipose tissue synthesis. What I'll do is these fatty acids and already the glycerol 3-phosphate, right? And then I'll have triglycerides. They'll esterify, and then I'll have increased triacylglycerol synthesis or triglyceride synthesis. So that's how insulin affect lipids, inhibits hormone sensitive lipase, which break down lipids, but it activates lipoprotein lipase, which break down blood lipids. Just think of them as blood lipids to get fat into my adipose tissue and increase triglyceride synthesis. For protein synthesis, insulin stimulate amino acid into the cells, the uptake of amino acids, and also protein synthesis, which is basically in your muscle. As we said, in system glucose, GLUT4 channels. And now let's talk about the counter-regulatory hormones against insulin. So basically, they do the opposite. Insulin decreases glycogen synthesis, decreases gluconeogenesis, ketogenesis, lipolysis, the breakdown of lipids. These things, they do the opposite. They increase glycogen breakdown. They increase glucose synthesis. They increase lipid breakdown and the formation of ketone bodies. Now let's talk about glucagon, which is one of the main counter-regulatory hormones. It is produced from your alpha cells in the islets of Langerhans. It is only one chain from 29 amino acids. You just know it's one chain. It has, pre-proglucagon and then proglucagon, and then after its release, it's only glucagon. It has short half-life, same as insulin, around 6 minutes, and it is a catabolic hormone. Okay, so it just tries to break down everything, and its main target is always the liver. What is glucagon regulation? Well, if I have high blood glucose and high insulin, that will inhibit glucagon. But when I have my catecholamine, so my epinephrine, norepinephrine, that means I'm in physiological stress. I want to increase blood glucose. They'll say, let's take our bro, man, the guy, the one and only glucagon, increase its stimulation and increase its secretion into the bloodstream so I can increase blood glucose. But also amino acids. So as you can see, amino acids increase insulin secretion, but also amino acids increase glucagon secretion. Why is that? Because when you eat a meal, right? When you eat a meal and then you have insulin secretion, insulin might cause extreme low blood sugar. It might cause like a lot of glucose uptake to the point where I have low blood glucose. So what the body does, okay, I have amino acids and that. What I'll do, I'll just increase glucagon a bit with the insulin to just try to cancel it out. So okay, I want the excess glucose to be taken up, but I don't want too much glucose to be taken up. So I'm gonna have also glucagon there to just prevent hypoglycemia. And of course, amino acids, because glucagon, it will increase amino acid uptake in the liver. Why? Because sometimes in your fasting state, so now all your glycogen is broken down, right? And you now need to just produce glucose. But from where do you get the glucose? Like, what is my carbon skeleton? From where I get all the carbons? What is the precursor? What glucagon does, it will increase amino acid uptake of the liver. So now there will be amino acids in my bloodstream. Okay, the liver will have increased uptake of these amino acids, and then these amino acids will be used in gluconeogenesis. So that's basically what glucagon does. Yeah. As you said, this is glucagon metabolism and its effect. Of course, glucagon, it uses the pKa, okay, so probably GPCR mechanism, but just increases the activity of protein kinase A. We've studied all this in MOL 1. As I said, increased glycogenolysis, gluconeogenesis. It also increases fatty acid oxidation, okay? And this is mainly due because when glucagon is high, like very high, right, insulin is very low. When insulin is low, what we'll have? We'll have activated hormone-sensitive lipase, and then I'll have increased lipolysis, and then these increased fatty acid oxidation. Okay, I'm gonna have increased adipose tissue breakdown, and these fatty acids will go to the liver, and then I'm gonna have increased fatty acid oxidation, and that what will cause? Will cause these oxidation of fatty acids will provide ATP, so it will provide energy for my gluconeogenesis. So there is no exact precursor, but what we'll do is just provide ATP for gluconeogenesis, because gluconeogenesis require ATP. And as we said, it stimulates hepatic amino acid uptake as they are used for gluconeogenesis. And now we go to hypoglycemia. Now, as we said, the normal level for glucose is from 70 to 100, 110 milligrams per milliliter, right? But if it's lower than 70, we have hypoglycemia. Okay, it's subtle, but it's not that much. But if I have the glucose level lower than 40 milligrams, that's severe hypoglycemia. And we have three types of hypoglycemia. Either if it's insulin induced, so basically maybe in diabetic patients, because I have a pump or like insulin, I just put the insulin secretions, right? And diabetic patients, insulin pumps. What if the pump malfunctions and it increases insulin secretion more than needed? So I now have high insulin. What will high insulin do? It will cause this severe hypoglycemia. And then I have postprandial hypoglycemia. So after you eat a meal, as we said, after you eat a meal, there is increased insulin secretion, but we also, what we need, some glucagon there, so it can cancel out the extreme glucose uptake. So, okay, excess glucose is uptaken, but still I don't want the blood sugar to be low, so I'm gonna have some glucagon to cancel it out. But what if the insulin is way too high that glucagon cannot cancel it? That what we call is postprandial hypoglycemia. And then of course fasting hypoglycemia. So if you haven't been eating for a long time, okay, maybe Ramadan, you know, أو أهلا في غزة, they might have fasting hypoglycemia because they have taken a long time since eating a meal, and unfortunately they'll have hypoglycemia, and that will cause big problems. Now, as we said, counter-regulatory hormones are the ones against insulin. They just try to do the opposite. We have glucagon, catecholamine, so epinephrine and norepinephrine, cortisol, and growth hormones. All of these are ERP. I don't know, I think they try to make it together, right? And they just basically do the opposite. Glucagon and epinephrine are mainly short term. Okay, so just for like short term, if I have physiological stuff, just at that time, I'm gonna release them. While cortisol and growth hormone, you know, cortisol is for prolonged stress. So that's what we have long term regulation. So glucagon, epinephrine, short term, while cortisol, growth hormone, long term. And you know, sometimes in diabetes, sometimes glucagon doesn't work. Okay, the alpha cells are also affected in some types of diabetes. So here, epinephrine is like your backup. Okay, glucagon is not working. I'm gonna take its place. I'm gonna increase glucose release. I'm gonna increase lipolysis, and I'll have enough blood sugar in my blood sugar. And yeah, now go to questions. Which molecule directly inhibits potassium channels in the beta cells of the pancreas, thereby contributing to insulin secretion? So first I have the glucose uptake by GLUT2, and then I'll have glycolysis, citric acid cycle, oxidative phosphorylation, and then I'm going to have high level of some certain molecule, which is ATP. Yes. And now, in normal states, which of the following activates both glucagon and insulin? So high levels of what increases both? What is it? It's amino acids. And now we move on to feed and fast cycle. It's going to be easier, inshallah. Moving on to feed fast cycle. It's just we'll talk about mainly three states, like when you're in fit state, and then a bit prolonged fasting, and the prolonged fasting, which is starvation. And then we'll talk about the adaptation of the four major organs: liver, brain, adipose tissue, and muscle. So let's start with the three states. So if we have first the well-fed, so immediately after a meal, so 0 to 3 hours post-meal. So this is after your iftar, you know, you just had your iftar until Taraweeh, so that's your state. And the post-absorptive state, you know, when it's like 9 o'clock, 10 o'clock, like it has been like 6 hours since your suhoor, and that. And then prolonged starvation state, which is way more than 1 day, can be days, weeks, which what we see unfortunately in our brothers and sisters in Gaza or in Burma, less. So, and now we need to study the mechanisms and the metabolism in each state. So first we need to know the major energy sources for carbohydrates. It's two ways: either blood glucose, okay, which mainly is used by RBCs. RBCs only use blood glucose; they cannot use anything other than glucose because they lack mitochondria. And then the brain, which mainly also uses glucose, but then in like starvation, it can shift to something else. And my second way of carbohydrates, like energy storage, is glycogen. You know, in muscle and liver, there is glycogen synthesis, there is glycogen storage, and that's the second way of carbohydrate energy storage. In lipids, fuel, it's mainly triglycerides and adipose tissue or fatty acids. But then in like fasting, these triglycerides are broken down to like fatty acids, and like in very prolonged fasting, we'll have ketone bodies, which mainly comes from acetyl CoA. And then proteins, we rarely use proteins, okay, unless it's like khalas, extreme starvation, because we really want to keep the proteins. The proteins are the most important. I just want to keep them because they are for my muscles, and without proteins can be big problems. So it's only used in extreme starvation, and it is paired like in starvation when ketone bodies are available. If they're not available, then there's the proteins. That's what you see in like very prolonged starvation. Some people, they don't have any muscle mass due to unfortunately that they have taken all the ketone bodies and now they're going for the proteins. And now let's see the hormone regulators. We have the insulin, only in fat state. It's very dominant when you just ate your meal. Glucagon in starvation and even extreme starvation. And catecholamines, cortisol, and growth hormone. With glucagon, they are the counter-regulatory. We have the epinephrine and glucagon, like physiological stress, and that, especially epinephrine. Cortisol and growth hormone are long-term. Glucagon is just when you have low blood sugar. And the regulation, now we need to know that insulin glucagon ratio is very important because they're always there, right? But there is a ratio. If the insulin glucagon ratio is high, that means there is high insulin, low glucagon. That means we are in a fat state, okay? And now we're trying to synthesize, we're trying to build up everything. But if we have a low ratio, so low insulin, high glucagon, then what I'll have catabolic pathways, I'll have mobilization of fat, I'll have breakdown of glycogen, I'll have glucose production, okay? And how do they do all that? Through allosteric regulation mainly. So they will this glucagon will phosphorylate this protein, which will cause increased gluconeogenesis while decreased glycogen synthesis. While insulin, what it do, it will dephosphorylate that protein and it will increase glycogen synthesis while decreasing gluconeogenesis. So they're almost opposites. And they also, of course, affect gene expression in the long term. As you said, RBCs only glucose. They cannot use anything other than glucose. But the brain, it uses also mainly glucose in fat state, even in like normal fasting states, but in prolonged starvation, okay, RBCs are the ones without the mitochondria. They don't have anything, right? So the brain is gonna be, I'm selfless, I'm not selfish, okay? I'll leave the glucose for the RBCs and I'm gonna use ketone bodies to spare the glucose for them. So now let's talk about the well-fed state. So this is when your insulin glucagon ratio is high. So what I'll have in the liver, I'm gonna have increased glycogen synthesis. Also in the muscle, okay? I'm gonna have now increased, as we said, glycerol 3-phosphate, right? And then this is glycerol 3-phosphate, and then I'm gonna have increased activity of what? Of lipoprotein lipase. So my VLDL, my chylomicrons are broken down into fatty acids. While what is inactivated? Hormone sensitive lipase. Hormone sensitive lipase is inactivated in this state, while lipoprotein lipase is activated. So when I have the fatty acids and the glycerol 3-phosphate, I'm gonna have increased triglyceride in this and increased adipose tissue. Also, I'm gonna have increased glucose uptake by adipose tissue and muscle because what? They have GLUT4 channels, okay? So they'll have increased glucose uptake. And of course, I'm gonna have increased protein synthesis and amino acid uptake. And for the red cell, uses glucose as normal, same as the brain uses glucose. And this is mainly, as we said, 3 hours. And yeah, this is basically it. And for the liver, actually, it's most of its energy is basically when you have excess amino acids, okay? The muscle has taken already enough amino acids. There is some excess amino acids. That when the liver oxidizes these amino acids, it gets energy, and that's where it mainly gets its energy in my well-fed state. Now in my early fast, so now at this time I'm recording this at like 11 a.m. So this is, I'm in this early fast state. So basically now I have the liver, right? The liver now, I have high glucagon and low insulin. So low insulin glucagon ratio. Now glucagon is higher. What I'll have? First, I'll have a glycogen breakdown. Okay. I'll have adipolysis because now there's low insulin, right? So hormone sensitive lipase activity is now increased. Okay. So now I'm gonna have increased adipose tissue breakdown. I'm gonna have increased fatty acids and glycerol. Okay, and they're going to the liver. And then also for the muscle, they're gonna start using fatty acids. At this stage, they use fatty acids mainly for their energy because there is not lots of glucose now. They're already, maybe their glycogen storage are almost done خلاص in the muscle. So now they start to use fatty acids for energy. And the liver starts gluconeogenesis now after glycogenolysis, producing glucose maybe from fatty acid, and then I have the fatty acid oxidation, which provides ATP for my gluconeogenesis. The brain still uses glucose. Red blood cells will, of course, still use glucose. So this is basically it. And I just wanted to notice there's something I forgot in the well-fed state. if the glycogen storages are full, okay, if the glycogen storages are full, خلاص خلاص I have enough glycogen storage and still have glucose in the liver. The glucose will then be used to fatty acid synthesis. Okay, so that's why people get fat basically, because they eat too much food. Okay, now they have a lot of glucose. Okay, they might be like, they say, okay, I'm not eating fat, it's keto diet, but then they are in a caloric, like a lot of calories. They're eating a lot of calories even though without fat. They have increased glucose, and this glucose is going to be used for increased fatty acids, for fatty acid synthesis, because glycogen storages are full, which will be used for adipose tissue. And yeah, this is basically it. I know I just went up, but yeah, this is basically early fast and post fast. And now for prolonged fast, this is when we're going for starvation. Okay, now I still have fatty acid breakdown. Okay, hormone sensitive lipase is working overtime, and the liver now, because I'm gonna have the gluconeogenesis, okay, is not that much. First, it will use, as we see, amino acids from muscles. There are some amino acids, not like the protein is not fully breakdown, just some amino acids are used by muscle for what? Gluconeogenesis. Okay, because glycogen storage خلاص are done. Now I need to use these amino acids for gluconeogenesis. Why? Because I need a carbon skeleton. I need all the carbons, so I'm gonna use these amino acids. But it's only certain. It's not protein breakdown, because here we have something called protein sparring. Because okay, now I have glycogen storage is full. Okay, I have fatty acids released, but then the brain cannot use glucose. This image is wrong because I couldn't find an image for prolonged fast. But basically, now the brain cannot use glucose because خلاص, it needs to leave whatever glucose we have to the RBCs. Fatty acids cannot cross the blood-brain barrier. Okay, it cannot cross it. So what we'll do something where we produce fatty acids, and then I produce acetyl-CoA, and from acetyl-CoA, I'm going to produce ketone bodies. Ketone bodies will then be used by the brain. Almost 2/3 will be used by the brain, and 1/3 will be used by glucose. Just a bit by glucose, but mostly is from ketone bodies. My muscle will now also use ketone bodies as energy source, just to spare the protein. It does not want to break out the protein. And of course, red blood cells will only use glucose. Now, the liver, why doesn't the liver use the ketone bodies? Because it lacks an enzyme called thiopherase, which is used for using the ketone bodies for energy. It's called thiopherase. And at this state, it's just using the fatty acid oxidation energy for it. And that's for my prolonged fast, as you can see. Yeah, this is basically it. Of course, glucagon and epinephrine is going to be high. These states. And yeah, also renal medullary cells. It was mentioned in the old lectures. I don't think it's now mentioned in this, but just keep it in mind. Maybe she asked about it. I don't know. But yeah, this is prolonged fast starvation. Now, as you can see, these are the energy sources that I just ate, right? The ingested glucose is done after 4 hours. And then the energy source mainly is going to be your glycogen storages. And after 1 day, when your glycogen storages are depleted, it's mainly going to be gluconeogenesis as the source of glucose, which has its carbon skeleton from the amino acids from my muscle. As you can see, these are the fuels, right? The metabolic fuels at the start of a fast of 70 kilogram man. He has 15 kilograms of fat almost. Okay, 6 kilograms of proteins, but it's not used a lot, and only 200 grams of glycogen, right? So the fat storages in the average man, okay, is sufficient to meet energy needs for almost 80 days. So sometimes people can go for 80 days without eating, but they'll probably die before that due to ketoacidosis or something like that. Now, liver roles, as we said, in fat state, okay, the liver uses mainly the glucose, the excess amino acid as energy. Okay, I have excess amino acids. It goes to the liver because it's the factory that filters everything. It will filter it and does everything for that and uses it as energy in the well fed. In the and then when in my post-absorptive state, in my start of fasting state, right? What I'm gonna use the fatty acid oxidation, and they produce lots of ATP, and that will be the energy source for my liver. And then at prolonged states, it's always still the fatty acids because they cannot use the ketone bodies as energy because it lacks thiopherase, which prevent it from using ketone bodies, as you said. And now for the brain, we said at well-fed and even post-absorptive, it uses glucose only, but then at prolonged fasting, at starvation, it wants to spare the glucose for the RBCs. So 2/3 of its energy is going to be from ketone bodies because it has the thiophorease, so it can use it as energy. Why not fatty acids immediately? Because fatty acids cannot cross the blood-brain barrier. And of course, the glucose enters through GLUT1, GLUT3. These are just like numbers, but I don't think she's going to ask about it mainly, just for your information. And then for adipose tissue, well fed, I'm going to have activated lipoprotein lipase, while deactivated, I'm going to have the inhibition of what? Hormone sensitive lipase. So now I'll only have adipose synthesis. I'll have triglyceride synthesis, right? I'm going to break down my chylomicrons, my VLDLs, and I'm going to have increased glucose uptake. I'm going to have now increased glycerol 3-phosphate and an increased fatty acids from the breakdown of the VLDLs and chylomicrons through lipoprotein lipase. These fatty acids and the glucose 3-phosphate together, they give me triglycerides, and that's in the well-fed state. Now, in the post-absorptive and the starvation phases, I'm going to have fat breakdown because there's no insulin now. My hormone sensitive lipase is very active, okay? It's very active. My lipoprotein lipase is deactivated at this state. So I'm now going to have adipose tissue breakdown. I'm going to have increased fatty acid release, increased VLDLs, and chylomicrons. As you can see, yeah. Of course, insulin stimulates glucose entry, but yeah, basically it. And then for my skeletal muscle adaptation and well-fed, it's mainly glucose, okay? Just glucose, and I have glycogen storages. And then the post-absorptive, I'm gonna start depleting my glycogen. And then if the glycogen storages are depleted, I'm gonna start to use fatty acids, okay? Fatty acids as my energy source. And then at starvation phases, because I don't want protein breakdown, at all. What the muscle will do, they start to use ketone bodies as an energy source. Now, yes, even in like post-absorptive stages, right, at start of the fasting state, I'm gonna have some amino acid releases for the gluconeogenesis in the liver. But you need to understand it's only a small proportion of the protein. It's not full-scale protein breakdown. It's just some amino acids, like alanine, that are mainly used for gluconeogenesis, that are released. But I'm not breaking down the whole proteins. It's just these amino acids that I release for gluconeogenesis in my fasting and starvation states. So yeah, so muscle, well-fed, glucose, fasting, fatty acids, okay? And then at starvation, it will use ketone bodies to spare the proteins and do not break proteins. And now for questions. Oh my God. During prolonged starvation, why is it loading? Problem sometimes with, okay, now it's wrong, okay. So during prolonged starvation, what will happen? Exclusively utilize ketone bodies. Hepatic glycogenolysis, so breakdown of glycogen is main source of blood glucose. Will blood glucose fall less than 40 milligrams? So now it's severe hypoglycemia. Will fatty acids be metabolized as a major gluconeogenic substrate? Or the energy demand of the body are met primarily by the mobilization of source of triacylglycerols, which are my triglycerides? This is actually a good question, you know. It's E. Now, I know the choices look so much. Why is it not correct? This one, because it said exclusively. In prolonged starvation, it's 1/3 glucose, 2/3 ketones. So that's why it's wrong. B, hepatic glycogenolysis is the major source of blood glucose. This is not correct because already the glycogen stores are depleted from my fasting state. The main source of blood glucose now is just gluconeogenesis, which is not produced from fatty acids. Fatty acids cannot be used as a gluconeogenic substrate. They provide the ATP for gluconeogenesis. Their oxidation provide the ATP for gluconeogenesis, but the actual substrate for gluconeogenesis, the actual carbon skeleton, is from what? My amino acids, my alanine, my that. It's not from fatty acids. and the blood glucose falls to levels less than 40 milligrams, that's severe hypoglycemia. I'll have major problems. What I'm doing, even if I'm prolonged starvation, okay, I'll still have my blood glucose try to be in normal levels, maybe a bit hypoglycemic, less than 70, a bit, but I'm not gonna have severe hypoglycemia, especially if I'm a healthy individual. Yeah, maybe I have gone 3 4 days without eating, but that's what's my body for. The body is now doing all this gluconeogenesis, all these things, just to maintain my blood glucose as a normal level. Because why? Because that's the level where RBCs can use the glucose. If it's way lower than 40, then some of my RBCs will not be able to use this glucose because it has very low concentration, and then I'm gonna have RBC death, which is gonna be detrimental. Okay, but the actual correct is yes, E, because I'm gonna have ketone bodies produced from triacylglycerol. I'm gonna have the fatty acid oxidation, okay, giving the energy for the liver. Also, in muscle, they will use the ketone bodies. So yeah, the most of your energy demands at prolonged starvation is from your adipose tissue. Now, glycogen stimulates gluconeogenesis and what during fasting? Gluconeogenesis is that the production of glucose. Is glycogenolysis, the breakdown of glycogen. Oh, glycogenesis is the synthesis of glycogen. I didn't know it earlier. But yeah, whenever you see synthesis, protein synthesis, fat synthesis, it's probably not production of glucagon. Now, glycolysis might seem because of the breakdown of glucose, but remember, I don't want to break down glucose. I want to produce glucose as it is. Okay? So that's a wrong. Glycogenolysis and gluconeogenesis, because glycogenolysis will produce glucose. And now, خلاص, we're done with the fit fast. And now for the last one, وأخيرا, we'll have diabetes. Okay, now finally for the last one, diabetes. Inshallah, it's gonna be easy. We're gonna go through it. It's not as hard as some people think. We'll go through it very easily. So, what is diabetes? We're talking about especially the most known diabetes, which is diabetes mellitus, which is basically, well, how do we define diabetes? High fasting blood sugar. Fasting blood sugar. Fasting is the most important thing because after you eat, yes, your blood sugar is gonna be high for a while. But if it's after like 5 hours and it is still high, that means there's problem with your insulin, which is diabetes. So what is the main damage by diabetes? Why do we need to treat diabetes? It's important because glucose is basically a poison in your blood. Okay, it is very oxidative. It can cause oxidative stress, generating reactive oxygen species. It can also cause osmotic stress, which will affect cells, and has something what we call AGEs, advanced glycation end products. It binds to proteins, okay, and then what it does, it will like break protein bonds, it will break proteins, it will denature them, which is very dangerous, especially if it's like essential proteins, maybe in the heart, maybe in your muscle, maybe in your brain, and that's very bad. So, and what we call them mainly like retinopathy, so the damage of the proteins in your eye, which will cause maybe blindness, or neuropathy, damage to your neurons. So that's how it works. Okay. So how do you diagnose diabetes? How we say, okay, you have diabetes? Well, there is one about the measuring the fasting plasma glucose. So after like the doctor will tell you, I want you to come after you have ate after like 12 hours. Okay. I want to measure your blood sugar. If your blood sugar is above 126 in two separate occasions, we can say that you have diabetes. Okay, because the normal is around 70 to 100, 110. But if it's higher than that by a while, as in fasting state, that means you have a problem. But it's not the main thing that we use. The main thing that we use, which is also long term, is my HbA1c test, which basically tests how much of your hemoglobin is glycated. As you remember, the advanced glycation end products where glucose binds to a protein. Now we're measuring how much of my hemoglobin is glycated by glucose. Oral glucose test is not that important. They don't use it. Like they see how much how much sugar is in your urine. If it's above 200 or higher,
then you probably have diabetes. But it's not like the golden standard. This is the golden standard, HbA1c. So we say that HbA1c is basically how much of your hemoglobin is attached to a glucose. If it's 6.5% or higher, that means you have diabetes. This is a test or a positive test for diabetes. If it's between 5.7 to 6.4, so 5.7 to 6.4% of your hemoglobin is attached to a glucose, that means you are in the pre-diabetic stage, and this is mainly in type 2 diabetes. Okay, if you catch in the pre-diabetic, that's where I tell you need to exercise, you need to lose weight, blah blah blah. I tell you why all these things, so you can avoid diabetes. But why do we use HbA1c? Because it's, as we say, اللي يضحك. Okay, protein, hemoglobin, and red blood cells have a lifespan of 120 days, 3 months. Okay, so let's say, for example, you haven't ate, maybe you are actually going to the doctor, you're always eating sugar, you're already pre-diabetic, right? You're always eating sugar and that, but then before coming to the doctor, you fast for 2 days. So your fasting blood glucose can be low. But this will not be effective. This will not be low because this is around 3 months. It tells you how much activity or how much sugar you're eating for 3 months. So that's how HbA1c works. Now let's talk about type 1 diabetes. That's the diabetes that we always think that's the genetic one, right? But actually type 2 is more genetic than type 1. Just keep it in your mind, okay? If they ask you which one has a bigger genetic determinant, it's not type 1, it's type 2. What happens in type 1? Yes, you will have some genes that is exposing you to may have type 1 diabetes, making you in more risk, okay? But what actually causes in type 1, type 1 is the autoimmune destruction of your beta cells in the pancreas. So your immune cells will suddenly start attacking your beta cells in your pancreas, destroying it, and then you'll have no insulin secreted. So tell me, how does it happen? Okay, basically, when you're young, maybe 2 3 year old, you will get exposed to a certain virus, a certain toxin that maybe other people that don't have that genetic predisposition that you have, they might have also been affected by it. But the difference between you and them is because of that virus or that toxin, you will have an immunological trigger. Your immune system will be triggered now to suddenly attack your pancreas. How? Don't ask. We don't. Okay? And the beta cells will not be destroyed immediately. It's going to be gradual. Okay? So maybe your immunological trigger while you are like 6 months or 2 years, right? But then it starts to destroy, and you'll have no symptoms at all because there's still insulin secreted because not all beta cells have been destroyed. But then after a while, when you reach prepubescent age or around puberty, that's when around 90% of your beta cells have been destroyed. And that's where the symptoms start. That's where the symptoms of type 1 diabetes start because now your insulin secretion is very low, that the glucose now is comfortably high in your blood, which will cause all these things. So around prepubescent puberty age, where you have the symptoms of type 1 diabetes. What are the symptoms, you'll ask? Okay, the symptoms are 4 things. Okay, you have polyuria, so excessive urination, polydyspia, which is excessive thirst, polyphagia, which is excessive hunger, and weight loss, excessive weight loss. So let's talk polyuria, because you'll have high blood glucose in your blood, right? The kidney will not be able to secrete to reabsorb all that glucose. So some glucose will stay in your kidney and then your urine, and that glucose will cause osmotic stress, telling all the water, come, reabsorbing a lot of water, which will lead to polyuria. And now you have lost a lot of volume, right? What will happen? Your body will start to be in excessive, start to be thirsty, because I have now volume depletion. I need water, so I'll tell you, please drink water. So that's why polydyspia, excessive thirst. But then also, if you don't have any insulin, right? What is the problem? That your GLUT4 channels in your muscles and in your fat will not be able to take glucose. Now, these things are major energy reservoirs. If they do not get glucose, what will they do? They'll basically tell your body, we are starving, we are in starvation mode. So the body is in starvation mode, although glucose is in the blood. We have the energy source. Why? Because basically this is what I call it like pseudo starvation. The muscles and the fats, they have the glucose, but they just there is no insulin telling them to take me in, take me in. So what it seems like there is no glucose. The muscle and fat send signals. We are in starvation mode. So that's what I call pseudo starvation. And if you're in starvation mode, you have excessive hunger. And also in starvation mode, you'll start to break down your fat, even your muscle, protein breakdown, which will cause your weight loss. Now, what are the three main metabolic changes in type 1 diabetes? As we said, first of all, the easier one is hyperglycemia, because there is no insulin, so your liver will still do gluconeogenesis, although there is glucose in the blood, but there is no insulin in the liver, there is. So there will still be gluconeogenesis rate in your liver. And your muscles and fats cannot take glucose, okay? So the blood glucose is gonna be super high, because we said fats and muscles are the main energy reservoirs. They're the one that store the most glucose. If they cannot store it, the glucose is gonna be very high. So first, hyperglycemia. Second is ketoacidosis. Why ketoacidosis? Because now, as we said, we're gonna be in this pseudo starvation mode. Fat is going to be broken down from adipocytes, okay, free fatty acids going to the liver, and then the liver will start what? Producing ketone bodies. Who will use it mainly in this case? Muscles. Because muscles now think there is no glucose, there is nothing, we are in starvation mode. And if you remember, like excessive starvation, what do muscle use for like main energy source? They'll start using ketones. And if ketones accumulate in your blood because there is high production of them from the liver, what will happen? You have ketoacidosis. They will lower the pH in your blood, and that will cause like insane problems. So second problem, ketoacidosis, hyperglycemia, ketoacidosis. Third one is triacyl hypertriacylglyceroma, which just means high amount of triglycerides in your blood. Why? Because and now in adipocytes there is no insulin, right? So your the insulin is not affecting the adipose tissue, so your hormone sensitive lipase will be very, very high. Okay, it will just break fats, and what will be low? Your lipoprotein lipase, which breaks your chylomicrons and VLDLs. Now, what will that mean? Now, I'll have high a number of fatty acids, okay, and glycerol. They will come to the liver. Some will be produced as ketone bodies, but others they will be returned into VLDLs, and then from what you eat from your testes will become chylomicrons. So these are basically triglycerides in your blood. And because there is no lipoprotein lipase breaking them down, because there is no insulin activating that enzyme, they will accumulate in your blood. So you'll have high amount of triglycerides in your blood, hypertriacylglyceroma. So the three main metabolic changes in type 1 diabetes is hyperglycemia, ketoacidosis, hypertriacylglyceroma, which is just high amount of triglycerides in your blood. As you can see, this is just an explanation. The percentage, I don't think she's gonna mention it, so you don't need to worry about it that much. Treatment of type 1 diabetes, as we said, because there's no insulin, right? There's no insulin pump. You just need to insulin therapy is needed. An insulin pump, insulin injections, it's needed. It's not like type 2 where maybe there's prevention. No, you need just injections of insulin now throughout your life. But there is a problem with the type 1 diabetes, and it's imagine if your pump malfunction, okay, or maybe you inject a high amount of units by mistake, right? That's when you get hypoglycemic shocks. Okay, so for example, let's say that I put a high amount of insulin in my body. I'm not diabetic, right? But what will happen? I'm not gonna have a hypoglycemic shock. Why? Because I don't have diabetes, I'll have what my counter-regulatory hormones. I have my glucagon, the epinephrine. They'll see that high insulin. They will be secreted now in high amounts in my body, preventing hypoglycemic shocks, and then they will cause the high blood glucose. So the insulin will not have that much of an effect. But in type 1 diabetics, because especially chronic diabetes, they'll have neuropathy, so their neurons will be affected, so epinephrine cannot be really secreted, they don't have the signals. Also, your immune system will not only break and destroy your beta cells, can also destroy your alpha cells. So there will not be glucagon released. So there will not be any counter-regulatory hormones there. So if insulin will work alone, they will lower your blood glucose to insane low amounts, giving you hypoglycemic shocks. So, okay, have you seen, like, especially on Instagram, when we say, "Oh, I injected myself for example, 12 units of insulin by mistake, so now he's eating every sugary thing in the world, so he just don't have a hypoglycemic shock." Now, let's go to type 2 diabetes. Type 2 diabetes is mainly two things. First, insulin resistance, okay? High insulin resistance, then beta cell dysfunction. So, first of all, like, insulin is secreted by your beta cells. There is no problem in your beta cells. But your muscles, adipose tissue, basically are less sensitive to insulin. Maybe they have lower receptors for insulin now. Maybe the receptors just there is a problem, they don't work. But basically, they don't detect insulin. Insulin is in the blood, but they just don't detect it. Okay, and what will that cause? That will cause a high amount of glucose. Now they still react to insulin, but insulin need to be in much larger amounts. Okay, so they have what we got this insulin resistance. It's not like they don't react to insulin at all now, no, but they just have lower amount of resistance, so insulin need to be secreted in much larger amount just to exert the effect in normal conditions of like 1 unit. So in type 2, like, you need 10 units of insulin to do the effects of normally of 1 unit. Okay, and that will cause, especially in long term, that will cause hyperglycemia. Okay, because there is not a lot of glucose being taken in by the adipose tissue and the muscle, causing hyperglycemia. Other metabolic effects like ketoacidosis or dyslipidemia or these things, they're much milder in type 2. It's not as big and of a problem in type 1. Type 1 is like happening in large scale, but in type 2 is not that much. Type 2, as we said, is the more genetic determinant. Type 2, do they need insulin therapy? Maybe after a while, if they have their type 2 diabetes untreated, right? And then after a while, what we call after high insulin resistance, there will be beta cell dysfunction. Basically, the beta cells being burnt out, saying, I cannot produce more insulin, khalas, I'll go on turn off mode. So now there will not be any insulin released. And that's when you need insulin therapy. And the symptoms for type 2 is mainly polyuria and polydipsia. So excessive urination and excessive thirst. And that's the most common one, especially in the report. As you said, in type 2 is the one that we have pre-diabetic. Okay, that's when you are at risk, but you can prevent diabetes. You can prevent it. And how do we define either 5.7 to 6.4% or a fasting blood glucose that is from 100 to 125? It's slightly elevated. It's impaired fasting blood glucose. And that's what we're telling you. You're pre-diabetic, please exercise, lose weight, and all these things to prevent your diabetes. We'll explain why. As you see, okay. Your insulin resistant risk factors. As I said, in type 2, you start to have insulin resistance. Okay? Basically, the main risk for insulin, as you for type 2 diabetes, as you've all heard, is obesity. Okay, when you're very obese, there is a high amount of fat percentage in your body. Why? Because first of all, now when you have high amount of fats, you start to have more pro-inflammatory secretions like interleukin 6, these things, and less anti-inflammatory secretions like adiponectin. Why is that important? Because adiponectin, anti-inflammatory secretion, they will increase insulin sensitivity. Now, when they're lower, there will be less insulin sensitivity, and that will lead to what? High insulin resistance. Also, now in type 2 diabetes, when there is like glucose insulin resistance and the muscles and the fats, they do not take as much glucose, they will also go into starvation mode. So you'll have a lot of what? A lot of breakdown of fats. And now muscle will start to use fats rather than glucose. Okay? They will start using fat rather than glucose, free fatty acids and these as an energy source rather than glucose. So now they start to not use glucose, so now glucose is even higher. Okay? And at the end, with a high amount of lipid metabolites, which are toxic, by the way, these can also go to your beta cells and they can affect them and destroy them, the lipid metabolites. Now, this is not inside the lecture, okay? What you need to know inside the lecture that after a while, lipids, okay, and free fatty acids will suppress glucose-induced insulin secretion. I'm explaining you why, because some lipid metabolites actually will affect the beta cells and destroy them. Okay, so these are the main things why obesity will are great risk of type 2 diabetes. Now, when you see in type 2 diabetes, as you see the normal guy, okay, they had the same blood glucose, so they had the same meal where their blood glucose, okay, after they just ate a meal is 140. But look how much insulin units did the normal person that is not obese need to secrete? 40 units. But the obese person needed to secrete 120 units just to have the same effect. So it needs to secrete 3 times more insulin just to have the same effect. So you could say that the obese person have 3 times less receptors in our muscle and in his muscle and his tissue to the normal person. So this is basically it. But then this is what I explained, and you have also steatorrhea because when you have high number of fats in liver, it could cause fat deposition in your liver, as you've heard, like fatty liver. This is how type 2 diabetes progress. As you can see, at first you have high amount of insulin secreted, right? But then, as I said, after a while, especially in chronic diabetes, if you don't like start to lose weight, lose this fat, start to exercise more, your beta cells start to be destroyed, either by the fat metabolites or being burnt out. I cannot secrete more insulin. And they'll be destroyed. And if it's left untreated, they will be fully destroyed. And then you need insulin therapy. So that's how type 2 diabetes progress. First, insulin resistance, you'll have high insulin secretions, okay? But then after a while, the insulin secretion is going to go low because of the beta cell burnt out. Yeah, this is the metabolic changes. As you said, it's much milder. It's mainly hyperglycemia, but like ketoacidosis, dyslipidemia, it's much less than type 1. Type 2 prevention, it's mainly BMI. Okay, lower the amount of fat in your body, and you're in a much better position to prevent type 2 diabetes. And now we have the 6 anti-diabetic agents. Unfortunately, you need to memorize them. Okay, but I think it's gonna be so. First of all, metformin, the drug that Dr. Ahmed Al Jada loves. Okay, I think if Dr. Al Jada can marry metformin, he will. Okay, so metformin, basically it decreases hepatic gluconeogenesis. Okay, we don't know the mechanism of action. Don't ask me, don't ask Dr. Al Jada, no one knows. Okay, it just decreases hepatic gluconeogenesis. Okay, so there'll not be a lot of glucose production, and that's what we use mainly for type 2 diabetes. We have also sulfonylureas. If you remember at the first, how insulin is secreted, that basically glucose comes, and then there will be high ATP in the beta cells. The ATP will close that potassium channel. Closing potassium channel will cause the calcium influx, and then insulin secreted, right? Sulfonylureas will basically, okay, you don't need the glucose to do all this shit. I'll close the potassium channels myself. I'll cause the depolarization, and then calcium will induce the insulin secretion. So they basically just bind to the ADP sensitive potassium channel, closing it, which cause insulin release. Now, what is key effects? They can cause weight gain and increase risk of hypoglycemia because there will be high insulin, so you might like gain weight. And if there is a lot of high insulin, you might have hypoglycemia. And then thiazolidinediones. I'm not read ever again. This drug, whenever you just see thiazo, خلاص, thiazo, or I'll call it thiazo, it basically just increases insulin sensitivity, decreasing the resistance in muscle and adipose tissue. Maybe it increases the receptor, something like that. But it can cause heart problems. Now, GLP-1 agonists, this is your Ozempic. You remember incretin effect that when you eat 100 grams of glucose, there will be these hormones that will cause even more insulin secretion. This is basically an agonist, so it acts like these hormones, and this will cause high insulin secretion, while also it will reduce appetite. That's why, but you see people how they who take Ozempic, why do they lose weight? Because they'll have much reduced appetite. Okay, so they cannot eat much. But actually, when they take off Ozempic, they gain all the weight. So what's the reason? Just be normal. more exercise, calorie deficit. Why all these things? And the DPP4 inhibitors, basically they increase the amount of these hormones, okay, GLP-1 and GIP. So it's not acting like the hormone, like GLP-1 agonist, but basically it increases its secretion. It increases the secretion of GLP-1 and GIP by the body, and that will reduce appetite and improve glucose control. And then you have SGL2 inhibitors. So basically you inhibit the channels that reabsorb glucose in your kidney. So basically it just tells you to excrete all glucose. خلاص. I'm done with this shit. Excrete all glucose. And that will cause mild weight loss and lower blood pressure because you'll have lots of volume depletion. And it will also decrease blood glucose, of course. And now for gestational diabetes, if 3 to 5% of all pregnancies, the mothers, they develop gestational diabetes, so they'll be a bit diabetic, so they'll have high blood glucose. And that if that high amount of blood glucose comes to the fetus will start to produce high amount of insulin. Insulin is an anabolic hormone, so he'll have high weight gain. He will be very big in his age. That's what we call fetal macrosomia, and that's actually a big problem. I know he looks cute, but this is actually a very big problem if not treated, because immediately after he's delivered, he's gonna we're worried about hyperinsulinoma when he has high insulin, so maybe he's gonna get a hypoglycemic shock, so we need to treat it immediately. Yeah, and that's the questions. Now let's see. 57-year-old man had type 1 diabetes, he has hyperlipidemia, retinopathy. Which of the following contributes to the increased lipids in blood? Okay, this was clear because there is no insulin, so the hormone sensitive lipase will be very active. Second one, 50 ophthalmologist, cataracts, blindness, was 180, hemoglobin A1C 10%, ketones positive, glucose positive. Which of the following indicates the blood glucose has been elevated over a period of weeks? So the one that will expose you, okay? Which one will expose you that, oh, you have been eating a lot of sugar in the last periods of time? It's your HbA1c. And we're done, guys. خلاص. You finished the crash course. Inshallah, good luck in the exam. Maybe you're watching it one day before the exam or a week before the midterm, I mean. Inshallah. فاركم النجاح بإذن الله. والسلام عليكم ورحمة الله وبركاته.